morning, everyone, and I hope that there will be more of you soon. So, hello that early morning. We will continue our workshop with the second day, and we start with the distinguished guest, and that's Alexandre Varnek from University of Strasbourg and from Hokkaido University. And he will open today with the reaction informatics. So, Alexandre, the floor is yours.
Thank you. Bonjour, konnichiwa. So I follow my affiliations. So it's a great pleasure to be here and to present my talk, especially at 8:45, you know, in the morning. So not all students are here, but the best are definitely here. Okay, so I will speak about kitchen today. Reaction from chemical reactions. You know, actually, students attending previous lectures, actually they have an opinion that, you know, as soon as they use predictive models and select in virtual screening or they generate a new fantastic structure, this structure will immediately actually be accepted by pharma companies, and a new drug will immediately appear on the market. But then, when this structure comes to the in hands of medicinal chemists, they say, okay, look, guy, no way, because either actually they don't know how to synthesize it, or the synthesis is too complicated. And what do we expect from chemoinformatics is to propose, let's say, simpler synthetic pathway. So, and actually the main hope is that artificial intelligence will accompany human chemists, and they all together, they will prepare actually all this synthesis. Okay, in fact, today I will try actually to discuss two questions. How practical are the existing AI tools for synthetic chemists, and can these AI tools replace human chemists in discovery of new chemical entities? So actually these two questions will be in focus of my talk. And but this discussion actually started many years ago between three Nobel laureates in chemistry, Alexandre, Todd, Robert Woodward on one hand, and Elias Corey on the other hand. And the first two stated that chemical synthesis is an art more than a science, and human creativity is well beyond the capacity of computers. On the other hand, Elias Corey, who was the father of retrosynthesis, disagreed with them, and he said that one day computers will outperform human chemists. Let's say today we will discuss who was actually right, and do we still need human chemists to synthesize something? And if today they are still needed, what happens tomorrow? Okay, let's discuss what actually we expect from AI tools. So we have a target molecule. The question is how to make. So we need to build up the synthesis planning, which delineates the pathway between starting material and target molecule. And this pathway could actually consist in several reaction steps. And then for each selected reaction step, we need to predict several important parameters. First of all, optimal reaction conditions. You know, the yield of chemical reactions depends on reaction conditions. So we need to optimize them in order to actually provide with the highest yield. Then we need probably to estimate thermodynamic and especially kinetic parameters, reaction rate, because reaction which you want actually to propose should not carry out for days. It should be fast enough. So in such a way, what do we need? We need to, let's say, build up this reaction pathway, reaction route, and for each step, we need to predict conditions and all these parameters, including reaction outcome. So this is a bit outcome of my lecture for today. So we'll discuss different applications of AI in synthetic chemistry, retro-forward synthesis, prediction of reaction kinetic yield, assessment of optimal reaction conditions, and then we will actually move a little bit forward. So we will discuss how data science could be combined with the chemical theory, and if AI can design completely new chemical transformations, and finally, what is the future of AI in synthetic chemistry? Go. Yes, just before start to discuss all this, I will speak about the important technology, important for us, because we have been using it for more than 20 years. We call this condensed graph of reaction. And in this technology, we condense all molecular graphs participating in reaction equations into one single graph, which is called condensed graph. And this graph is characterized by conventional chemical bonds, single, double aromatics, and such called dynamic bonds, characterizing chemical transformations, single to double, created single, broken double, etc. They are shown here in color. And this object could be considered as some sort of pseudomolecule for which we can generate smiles, fingerprints, descriptors. So we can, in machine learning exercises, we can work with chemical reactions as with the individual chemical structures. Okay, now I will start with retrosynthesis, actually suggested by Elias Corey, who this is pathway backward from target molecule to the starting material, which we call building block on our terminology. And if this pathway is successful, so it outputs this. So target molecule in my slides is highlighted in blue, starting material in green, and intermediates, precursors in rose. And then in retrosynthesis, the program is supposed first to fix such called strategic bond, to be disconnected, and then find the optimal pathway here, because this is a combinatorial explosion, because we have many different bonds, and these bonds are disconnected according to such called reaction rules, extracted from databases. And in such a way, we have this, not only one, but several pathways of this type. So far, people actually developed many interesting tools, both commercial and open source. They are listed here. So I will speak mostly about our development. Certainly, everyone speaks about his own development. So I will speak about SynPlanner tool, recently developed in the University of Strasbourg. And the main developer is Tagir Ahmedshин, our former PhD student. So this SynPlanner, compared to its competitors, is very convenient end-to-end tool which can be easily retrained on the reactions of the, let's say, particular laboratory or company, so easily customized. And actually, it learns chemistry using reaction databases. Database, and as a building block starting material, we use the commercial catalogs. now I have small demonstration how actually it works. So you have very simple graphical user interface. You can draw with a sketcher your chemical structure, target molecule, and then you push just, there is a setup which you can change if you want. Actually, what you can change the maximum number of reaction steps. By default is a 6, you can go till 9. And then you just push the button. This is actually developed for synthetic chemists. This is one-button software. You just push the button, and it gives you solutions. So for this molecule, actually, it has found more than 200 synthetic pathways. Just just to shorten a little bit this presentation. Yep. Oof, no. Actually, look, it lasts 3 minutes. Just believe me, it works. Actually, it can be downloaded from our GitHub. I advise those who is interested in, please download it, and we prepared a lot of tutorials, instructions, etc. Pretty convenient tool. Okay, anyway, I would like now to switch into the questions. How are reliable these tools? So what we did, we applied several different algorithms, of search algorithms. The most popular is Monte Carlo tree search, and then there are some analogs of this algorithms, some developed and implemented by my postdoc in Hokkaido, Milo Urkerol, and we applied all these algorithms in order to find synthetic pathways for 4 categories of molecules. So we selected from the Campbell database, we selected 400 molecules of different, and we split them on the bins, each corresponding to very important parameters measuring complexity of chemical complexity, which is called synthetic excitability score, developed by my good friend Peter Ertl. Yeah, he will speak in his lecture more in details, but large SA score corresponds to complex molecules, and the score about more than 5, it's already close to the score of natural products. So what you see, there is some small variation of as a function of different algorithms of search, but anyway, the performance of the software drops significantly with complexity. And this is a situation not only with our tools in Planner, this is a common situation with all retrosynthetic tools developed so far. So this is the main challenge of retrosynthesis. I wouldn't like actually to speak. If you have some questions, we can discuss it, but this is situation, So either the tool actually proposes you several hundreds or even thousands different solutions, or nothing. This is a bit, okay. Then now I would like to speak about forward synthesis, which in certain sense is even more advantageous than retrosynthesis. So this is a way from commercially available building blocks to target molecules or its analogs, and close analogs which predicted as a potent molecules. So what is important that here we can fix first of all, the limited number of steps, and select only such called robust chemical reactions, reactions which actually always work. So the key point here is that we are not obliged to find this pathway for the target molecule. We actually can stop at its analog. Okay, But here we have I have a few slides from Gisbert Schneider, so who actually developed one of the first tool for forward synthesis, which is called DOGS. And he mentioned this problem of combinatorial explosion when you combine many building blocks. Here he used, to my memory, 26,000 building blocks, different building blocks. And so if you consider all combinations, you can actually have too many, let's say, computing. So certainly people use different algorithms trying to optimize this. But anyway, so far, it's pretty long calculations. Compared to retrosynthesis, for retrosynthesis, we have a time limit 2 minutes. For this kind of calculations, Gisbert Schneider, even now, he actually spends few days. You know. But more than 25 years ago, he published first success with a scaffold hop. So this is his photo, Gisbert Schneider, who is a professor at ETH in Zurich. And he reported already first molecules with new scaffolds and highly active. And I would like actually to show you another slide related to relatively recent publication by Bartosz Grzybowski, who used this approach, forward synthesis, to demonstrate that the prebiotic molecules could be spontaneously synthesized from very simple molecules. You know, he discussed in his paper in Science, he discussed the possibility of spontaneous formation of life molecules in our planet. So in such a way, from all these starting molecules, which, you know, existing, which were formed upon the formation of our planet, actually spontaneous chain of reaction could lead to synthesis of these, you know, prebiotics. So I wouldn't like now to discuss this philosophic question of creation of life, because it's a bit dangerous. Catholic Church actually doesn't actually completely agree with Bartosz Grzybowski with respect to life creation on the planet. So I will turn to another interesting approach, AI approach, reported by Connor Colley. Connor Colley is a rising star in MIT in Boston. He's professor there, published many interesting paper in reaction informatics. And this is one pretty old paper where he developed a model, AI tool, predicting product for the given set of reactants. And he managed to predict not only one product, but also major product and also minor products here. And then what is interesting is, like, by the fact that Peter did with his SA score, he made a survey from, you know, synthetic, he organized some sort of competition AI against human chemists, suggesting actually either chemist or AI tool to predict products of chemical reactions. And he considered first two groups of chemists, all chemists, or let's say professors, best chemists, and different kinds of reaction. So very popular reactions, actually people can easily predict the product, or rare reaction, which actually is a bit more difficult case. And here you have human best in blue, which more or less overlaps with the model performance, and it's outperform human in average. So this is interesting result, which shows that AI tools are already competitive with the humans. You know, I remind you that all discussion between 3 Nobel laureates. Okay, now I would like to speak about reaction conditions. This is extremely difficult object to be modeled. Why? Because of the availability of experimental data. You know, organic chemists never publish negative results. They publish only successful results, only positive results. But for machine learning, we need both. Okay? Then another, so, you know, the one chemical reaction may proceed successfully under very different sets of reaction conditions. What reaction conditions? Solvent, temperature, catalyst, some additives, etc. You know, so it could be poison, catalyst poison. It could be acid or basic medium, etc. So recently we discussed in all the problems of modeling of reaction conditions in the perspective paper published in Chemical Science with our PhD student, Matthew Bolt, who is interested could look at. But generally, anyway, you know, despite of the difficulty, experimental conditions could be modeled. And there exist two major approaches to model it. The first one is similarity-based assessment, which is based on the main assumption then that similar reaction proceed under similar conditions. It looks similar. Do you remember basic similarity principle in drug design? So similar molecules possess similar biological activities. The problem is how to assess this similarity. It could be based on fingerprints, it could be based on pharmacophore, etc. But here, for chemical reactions, the main problem, how to assess similarity of chemical reactions. So I'll speak about a bit later. And then, the idea is very simple. So we have query reaction, so reaction for which you want to predict optimal conditions. And in the chemical space of reactions, you find the close neighbors. Okay? Or alternatively, you can build machine learning models. And we expect, so we have as an input reaction equation, a condition as an output. And we need to actually to provide with several alternative solutions because any chemical reaction can proceed under different reaction conditions. So just to show how it works, first let's consider a case study of a hydration reaction, which as a function of condition may lead to different products A, B, or C. You know, A means that this is a reduction of NO2 group to NH2, B, a cleavage of CO bond, and C both. And the idea is to, the goal is to find conditions which selectively lead to product A. Despite of, you know, this simplicity of this reaction, something plus H2, this is a pretty challenging task because people use very different metal catalyst, very different solvent. They can change temperature, pressure, etc. So we, for the modeling, we selected more than 200,000 reactions from reactions database, and then we discovered enormous problem with the data. Only a small part of them, less than 40,000, were annotated with temperature, pressure, and catalyst. So it means that even reactions database, which is considered as one of the best databases, existing databases, actually it is not, the data are not completed for chemical reactions, at least. Okay, anyway, then I'm very pleased to show a photo of my close collaborator, Timur Majidov, who was team leader at the University of Kazan. Now he's he's working for Reaxys, by the fact, in Oxford. And actually, with him, we decided to, let's say, use, first of all, for input, we decided to use descriptors generated for condensed graph. So condensed graph, just to remind you, actually it's pseudomolecule which encodes the entire chemical reaction equation. And for the output, we propose to present the output as a bit string in which we have 3 bits for temperature, low, medium, high, for pressure, low, medium, high, and presence catalyst, acid, base, and 1 hot encoding for the catalyst, metal catalyst. So altogether, so we have 29 bits for catalyst. It means that these are different metal catalysts. So, and in such a way, the neural network predicts the probability to be filled in this, how to fill this bit string, and then certainly we should take the high probability temperature or pressure, etc. So in such a way, we condense all this in shorter vector, from which one can calculate such called likelihood. And so for all possible combination, temperature, pressure, catalyst, etc., there are more than 2,000 different combinations. We can associate to each combination a likelihood parameter, and then we can rank all solutions according to these parameters and select first 3, first 5, or first 10. Okay? Or alternatively, in similarity based, as soon as we encode chemical reaction by one pseudomolecule, we can generate descriptors, and in this space of these descriptors, we can actually calculate similarity using Tanimoto index, for example. Like for the molecules, we can calculate similarity between the query reaction and database reactions. Okay, so this is like this, yes. So we can calculate distances and first take first 5 or first 10 neighbors. And what does it give? Actually, it gives reasonable results for, let's say, 10 first neighbors. Among first 10 solutions proposed by the model, we can observe the experimental one with a probability of 85%. Not ideal, but this is the best what actually people could do. So we compared this, we benchmarked this with the alternative models like Connor-Corey. It's roughly turns around this number. Okay, and also we did by the fact experimental validation. This is interesting. So we used H-cube from Thales Nano from Budapest, and 6 chemical reactions of this type. And for each reaction, we made a blind test. And then results are that we were successful predicting 100%. So this is experimental value, 100% yield. We're relatively successful for 2 other reactions predicting, let's say, moderate yield. And for this reaction was predicted successful, but it resulted in 0 yield. But this reaction was out of model applicability domain. But okay, so 1 out of 6 unsuccessful, not a bad score, I would say, for machine learning models. Now, I would like to switch into the yield modeling. Yield is the most difficult parameter to predict because we have many factors which affect yield. You know, you can lose your material during purification. The yield depends very much on the quality of purity of starting material, which you can hardly control. Actually, the experimental condition cannot be optimal, and simply human mistake. It's a very often situation. Also, we need to think about solubility, side reactions, and purification technique. Even the human chemistry has let's say, a lot of experience, choice of purification technique is very important. So, I would like now to report you results of a very recent study, not published yet, will be published soon, in collaboration with Ukrainian colleagues from who belongs both to University of Kiev, Taras Shevchenko University, and to Yenamin company. Yenamin mentioned yesterday as one of the main compounds supplier for medicinal chemistry. So, in fact, we used their in-house data, more than 3,000 yield values for such called Biginelli reaction. This is a reaction which is very good for combinatorial chemistry. There are 3 reactants, each bears 2 substituents, altogether 6. You can play with all of them. And they, according to, let's say, their estimations, for repeated experiments, they have a, let's say, standard deviation for yield about 15%. Okay, and all these reactions were synthesized according to identical experimental protocol, but by different people. Okay, so and we made 2 types of models: one regression model and another classification model, SVM model, and we achieved reasonably not bad performance for classification, around 0.8, but very bad for regression. In cross-validation, we had only less than 0.4, which is not actually acceptable. And then when we looked at the data, we discovered some significant problems. I don't know if there are some chemists in the audience, or you're just pure machine learner. But if you look at these 3 molecules, for this, we predicted 57% of yield, and experimental label was 0, okay? And then we found its close analogs in the training set, and these molecules, the products, actually differ from the first one only by position of one methyl group, which is pretty far from reaction center. And the experimental value for yield were 44 and 83, so. And here 0. Certainly, this is a problem. One can hardly believe in this. So, and then in such a way, we discovered more than 70 such cases, and we asked colleagues from Yenamin to resynthesize them, and they did. And then when we look at the resynthesis results, so this is a root mean square error in percent. For initial model, we had 50%. It's much larger than 15%, which they considered as intra-laboratory error. And after the synthesis, this error actually decreased to 30%, which is good. And then look for these 3 objects. We have experiment initial label 0 or very small, and then after the synthesis, it increased. And then close analysis shown that actually this is related mostly to purification issues. So in such a way, yield can, even if the human chemists follow the same protocol, actually, 2 different students, because many students from Kiev, they are working on Yenamin, actually they used a bit differently. Especially its purification step is very sensitive for this. Moreover, if, let's say, we try to generalize the situation with yield prediction, so what there are some data, new data reported, obtained in fully automatized experiments without no human intervention. And then people try to use data obtained experiment automatically as a training set, and then validate them on literature data or manually obtain intra-laboratory data. And results are pretty negative, in cross-validation for fully automated data, fully automated and fully reproducible. So they had fantastic results. Determination coefficient around 1. And when they tried to validate on literature or ENL data, actually they failed. So this is indication that we can hardly trust human obtained yield values, and the only way to model data is to use fully automatized setup. And so we need to combine in the training set only the data measured strictly at the same reaction conditions. Okay, now I would like to speak about a very interesting topic. If you're not tired, but anyway, you have no choice. I would like to speak about synthesis of new enantioselective catalysts. So this is a large molecules which mimic behavior of enzyme and which allows to from racemide, to mostly synthesize only one of enantiomers. Okay, and this development of this kind of molecules actually was the subject of Nobel Prize 2021. Benjamin List and David Macmillan, they got their Nobel Prize. So from the machine learning perspective, what we measure enantiomeric ratio in percent or delta G. This is difference between free energies of activation barriers, because this is all these reactions are proceed on the mostly kinetic control. This is a slide figure taken from the Benjamin List paper, which shows that these are catalyst, family of catalysts, ADPI, developed in his group. So actually the catalyst creates a binding site, like in protein, and you can control shape and size of the binding site by substituents, different substituents in different positions. I would like to give you details. Anyway, you can synthesize many molecules like this with a controlled shape and size of the binding site. Okay, and in experiments, so usually many entire family of this catalyst is applied, also one by one, certainly, to several chemical reactions. So we need to consider pairs catalyst reaction, not just one catalyst in one reaction. We have many of them. So, and in such a way, we need to, for machine learning tasks, we need to prepare the descriptor vector, which accounts for chemical reaction. So we used condensed graph of reaction for this purpose. Then catalyst, so we can use 2D or 3D descriptors. Then actually all these experiments proceed under different conditions, different solvent, different temperature. So we need to account all this. So prepare pretty complex descriptor vector. And we applied this technique successfully in collaboration with Benjamin List. In fact, Benjamin List also has laboratory in Hokkaido, so, and we collaborated with him as two different Japanese groups. Benjamin List and myself, actually, we were both played role as two Japanese professors, and these are colleagues who participated in this project. What actually we have done, we built the models on this is so we combined by the fact two different types of reactions, synchronization reactions, and carried out under different conditions, and the best catalyst in this in-transit training set actually provide with the enantiometric ratio 81 to 19 in percent. And then we built the model. Then this model was applied for virtual screening of a generated small library. And then we selected hits. These hits were synthesized. And we succeeded actually to design a new catalyst with much higher enantiometric ratio. Yes, okay. 92 to 8. Okay, and then Angivante even published a special paper, Varnek collaboration in Hokkaido. Nice. Okay, now the last part of my talk will be actually a bit more philosophical. So first of all, let's look at the future and discuss the question about creativity of AI. Because you know, machine learning models, they are not creative. So they extract knowledge from existing data and then they apply this knowledge. They can hardly actually go outside of the training set. What about newer AI tools? So we performed interesting work with the help of our PhD student in Strasbourg, William Bort. And what we did, we simply used smiles-to-smiles autoencoder. So we generated smiles for condensed graphs. So we trained it. And then we built with the, so we reduced dimensionality. And we sampled, we used color code to identify in the latent space of our autoencoder the area populated by Suzuki reaction. And we sampled from this area, autoencoder, hoping to find new types of Suzuki reaction. And you know, Suzuki, Akira Suzuki, by the fact he got Nobel Prize for his discovery. So, you know, the idea was, if, let's say, AI suggests us new types of this reaction, maybe actually together with AI we also get the Nobel Prize. You know. But then we discovered new, compared to the training set, new 13 new reactions detected, and then 5 of them have been found in recent publications. There's already success. It means that it can create something, really. It can create something new. But then when you're, let's say, you know, look at the message in red, you know, AI rediscovered known but untrained chemistry, purely from its imagination. But actually, this imagination, if you look at the results, is pretty limited. Because if you look at the eye of a synthetic chemist, the first example, this is red dot is found in literature, a green suggested by AI. So first actually is related to exotic coupling. Suzuki is a coupling reaction. So this is simply bromination reaction, you know. And another one, this is the CC coupling, but with a fluor, which is exotic living group. So we didn't get Nobel Prize for this, for sure. But anyway, we have, let's say, but is it really new? Can we speak about creativity of AI? I would say that not yet. And the main reason, compared to human intuition, the human intuition is based anyway on the basic concepts, mechanisms, chemical reaction. We can generalize chemistry. And purely, the model built just on the data is not able, actually, is not creative. So we need to combine this data-driven model with some fundamental knowledge of chemistry, physics, material science, etc. And now I would like to discuss this, how actually it can be done. So if, let's say, we consider, on one hand, on one side theory and on the other hand data. So we have two types of models: top-down theory-driven approach. For example, Schrödinger equation, which is a very nice model. It is not actually a ground truth, but it is very nice model. And we have let's say neural networks. It's purely data-driven approaches. And the idea is to combine them, to build up physics-informed machine learning, in which a fundamental knowledge, fundamental theory is accounted for in different perspectives. So either we actually insert fundamental equation directly in machine learning training procedure, or we simply build machine learning on quantum mechanics data, if you consider quantum mechanics as a fundamental science. Okay, let's illustrate all these two approaches. So first, I spoke about the importance of modeling of kinetics. So we build models on kinetic parameters of cycloaddition reactions. So these are different cycloadditions: 2 + 2, 3 + 2, and 4 + 2. 4 + 2, this is Diels-Alder. And then we manually compiled by the fact PhD thesis in Kazan University. So all students were working on this database, compiling values for log K, reaction rate, activation barrier, and the pre-exponential factor measured experimentally. Okay, and all these parameters are linked by the Arrhenius equation. And then what we have done with the help of our PhD student, Dmitry Zankov, we built two types of models. In the first type, all models were conventional. So loss function included just square difference between x and predicted values. We built individual models for each of these endpoints. And then we built such called conjugated neural network, in which all these three endpoints were predicted simultaneously, and we inserted Arrhenius equation directly in the loss function. Okay? By the fact, in sense of performance of these two types of models, in sense of RMSE, etc., the two models performed similarly, but Arrhenius equation was respected only for conjugated models because actually we insisted on this. Okay? And in fact, from the practical point of view, why do we need it? So let's suggest that we need to predict reaction rate at extreme temperature, which can hardly reachable experimentally, let's say near the freezing point. So we took this particular cycloaddition reaction, and then we applied conjugated model. Conjugated model, the trend as a function of temperature practically coincides with the ideal line. And if we predict all parameters individually and then apply for these predicted parameters Arrhenius equation, it leads to the value which is 2 orders of magnitude smaller than, let's say, a correct value, ground truth. So it means that already here, incorporation fundamental equation allows us to have much physically reasonable results. So another example is related to activity of my group in Japan. So we worked in close collaboration with Professor Satoshi Maeda, who is director of institute, and who developed a method which is called AFIR. This method actually allows to generate a sequence of chemical reactions, and the idea is to find a new chemical reaction generated with the help of his approach. And this is a DFT-powered search because energies are calculated in quantum chemistry. Okay, so what we did, we built the neural network which to model potential energy surface. And then we applied this in different case studies. So we actually apply this model to generate for the for chemical reaction, which proceed in the binding site of the catalyst, altogether more than 200 atoms. So we generated more than 20,000 equilibrium state and more than 45,000 transition states. You know, this huge amount of calculations. And then, let's say, ab initio, we reproduce experimental data. The yield of the two enantiomers, which the values are very close to experiment, and this actually is built practically can be considered as ab initio calculated results. Okay, now then, you know, I know that there are some quantum chemists in the audience, and actually typical for quantum chemists, if let's say size of the system is large, to replace bulky substituents simply by hydrogen atoms. Okay, so and this may lead to some controversy, as actually shown here. So there is a reaction which is called Passerini reaction, which proceeds in two steps. Alpha addition and then rearrangement, and actually it contains more than 60 atoms. So quantum chemists actually used model system, and then they observed some controversy because for a real reaction with real substituents, reaction rate is first order for each of reactants, whereas for this model reaction rate is second order, because at this step we need two acid molecules. So two molecules like this, they are needed for this rearrangement. And actually, when what we can do with our neural network, we can handle a real reaction. We don't care how large are substituents, because the neural network actually calculations are 1,000 times roughly faster than quantum mechanics DFT. So, and then simply we shown that there is no controversy. It depends on substituents, you know. For in real system, alpha addition is a rate dependent step. And whereas in model reaction, substituents are replaced by hydrogen, this is rearrangement. So it means that this is a very simple example, which shows that unfortunately, we can't actually be limited just by model objects like here. So we need to consider system as is. Okay, now I have still 5 minutes, and I would like to discuss actually, you know, so for the moment we use AI tools, just to conclude, as a collaborator, let's say. So we must ask computer to do something, okay? But actually, so what do we expect? We expect to have a decision maker which control fully automatized process of design and optimization of synthesis, new molecules. 12 years ago, Nature magazine published futuristic paper about robochemists, which could design 1 billion molecules on demand. It was related to a very popular in UK at that time project, dialing in molecules. Well, molecules you can order by phone. Okay, that time it looked like a fantastic, but you know, exactly the same year, Gisbert Schneider from Zurich, he published synthesis of potent molecules which were completely computer guided without any human intervention. So he used a very small device with containing two platforms, chemistry, which in which click reaction between two components actually proceed, and then bioassays, and then biological activity data actually went to the computer. The model was updated, and model guided the selection of new building blocks. So new compounds were selected, synthesize, etc., etc. And in the end of this loop, he managed to synthesize nanomolar molecules, okay, were predicted and experimental values were very close. So it means that, he started to do this, he went for the lunch, and then coming back, he found this. Fantastic, especially for PhD students. Yes, now, robots are everywhere. I wouldn't like to speak too much, this is yeah, it can, the robots can work 24 hours a day, and this is a video actually from the Nature magazine website for optimization of catalytic system of hydrogen synthesis from water. Well, anyway, what is important, but this equipment is extremely expensive. It is actually dozens of millions of dollars, in hundreds sometimes. But nowadays, actually, robotics and AI tools, they are actually becomes very popular, much more popular because of some sort of democratization. So, first of all, this actually, these are screenshots from 2 papers published last year when actually people reported very inexpensive robots which could run more than 12,000 reactions a day. 25,000 dollars are affordable for any even academic laboratory. It's not millions, only, Then, NVIDIA announced a newer supercomputer of low price. So we already have 2 of these in the lab. Fantastic. On which you can run not only, let's say, machine learning, but also you can train large language models like ChatGPT. And then, therefore, actually, everybody now is working on such called agents, which could be associated with ChatGPT or any other large language models in order to, let's say, execute different tasks automatically without actually reading manuals of computer programs, etc. So we did it also in the lab, and we have, let's say, I have no time, to believe me that actually you can just launch a prompt. How to synthesize something, so apatinib, okay, and then, okay, so a few seconds, it launches SynPlanner and it outputs results, and then it ranks the synthetic pathways, etc., etc. So it does it fully automatically. You just send a prompt, and this is extremely popular. Now this is actually taken from Wikipedia. This is the performance of AI compared to human in different tasks, and you see now with respect to reading, image recognition, language understanding, etc. AI it outperforms humans, and what we can expect that human could be excluded from this conversation, and instead of human, actually people already started to speak about general AI, who actually will give the order to agents instead of human chemists. And in 2050, actually, according to some estimation, this is a year in which AI will be able to perform any intellectual task a human can perform. And I don't actually exclude the situation like this in all modes. Then there's this audience and speakers will be there. But this is a bit perspective, long-term perspective. But today, I would like to finish this with this slide. Educational comment. It is not that machines are going to replace chemists today. It's that the chemists who use machines will replace those that don't. So, and I believe that in this case, the perspective of the students sitting here is very brilliant. You can actually, you know computers, and you can compete on the job market with those who don't use. So, acknowledgement to my colleagues from Strasbourg and from ELIXIR-CZ, Hokkaido, and then information about the next summer school in Strasbourg, which will be held as usually last week of June. So please welcome, registration is open. And thank you very much for your patience, for your kind attention.
Thank you for your talk. I would like to ask concerning your statement that AI has rediscovered some known chemistry, but which on it was not trained. I would like to ask concerning how many prediction it made, that the prediction was simply made by chance, simply by combining many possibilities, and from those you simply randomly rediscovered something interesting?
In fact, we used, let's say, we used text-to-text, well, smiles-to-smiles autoencoder, and actually it is not randomly. It samples. It evaluates the probability, you know, for to add a new symbol after the at certain level. So it means that it is based on he learned anyway, you know, the symbols order, and most of new smiles which autoencoder generated actually were very similar, were in the applicability domain, but few of them were a bit out. I wouldn't say this is cultural revolution. It's a bit out. It calculated simply probability, and probability was not zero. It was based, it was not random. It was based on certain experience. Okay? So, but it was outside of the area of training set, occupied by training set, but not too far, you know. We always we see this in machine learning models that, you know, even for molecules which are outside of ID, predictions could be good. But anyway, the distance between these molecules and the area populated by training should not be too big. You know, it could move a little bit outside, but not too far. So we need to generalize. We need to, let's say, to teach AI basic, let's say, fundamental knowledge. We need to give fundamental knowledge about what people already have generalized, the mechanism of chemical reactions, you know, all this concept from theory, you know, frontier orbitals, etc., etc. Still sleeping. Thank you. Thank you.
Okay, good morning again, this time with my lecture, and I will be speaking about the field of alphafoldology. And that's basically the term we coined together with Marian Novotny in 2021. And we coined it in order to describe that we now have a world where we have a model for basically any protein. And the reason why we need that is that still in 2026, we still don't have experimental structure for all human proteins. We have mere 40% of the structures of the human proteins experimentally known. This doesn't change much from the 2024. And in total, we have basically 250,000 protein structures deposited in protein data bank. And that's much smaller number than the amount of the proteins which we have in UniProt. That's approximately 200 million. So several orders of magnitude bigger number. And if we looked on metagenomics, so if we basically sequence anything which is in a dirt or in a pool, then we get metagenomics, and the metagenomics is even bigger. That's over the billion of the sequences. So there is a huge gap between how many sequences of proteins we can have and how many structures we experimentally know. So in order to get that, there is a prediction of the structures, and the prediction of the structures is governed the field. Basically was trying to find out how it's scoping up by via the critical assessment of structure of proteins, which is still a task where every 2 years the experimentalists give their structures to the organizers only, and the protein designers and protein structure predictors have only the sequences, and then they are trying to predict the structures, and those two things are compared afterwards. And as you can see from the early beginnings, if you had easy target, which means that you had a target which has a template for it, so basically the same protein but from different organism, let's say, it was relatively okay-ish even at the beginning of that CASP competition. But if you were looking for a protein which was novel, so there was nothing similar in a protein data bank, which is, then you can see that till 2016, there was basically really a limited advancement, and especially since 2006 to 2016, it seems that basically it flattened. Then in 2018, a new first company, which was DeepMind, joined the competition, and they were better than the rest of the field, and that's this orange one. But it was also a controversial as well, because all the academic labs had that models open, so everyone else was able to build on what they did. And DeepMind said, no, our model is closed source, and we have the Google computation, so there is no academic lab that would have enough computational power as we had in our premises. So for that reason, of course, the scientists were not happy, let's say. And in 2 years later, the academics actually were better than it was the DeepMind 2 years prior. But at that time, DeepMind came up with the AlphaFold 2 model, and the AlphaFold 2 was okay at all stages, and that's the worst case scenario. And you can see that basically even this model is still, the fault is okay, and the only thing which is different is that there is some movement in the structure, which of course can happen. So since 2020, there were plenty of newspapers like the protein folding problem is solved and everything like that. And the problem was that it was still closed. What happened later was that they've opened it in July 2021. And since they opened it, and they opened it both open source to the AlphaFold 2 source code, and they've also calculated 200, well, at that time they've calculated just the model organisms like human or mouse or whatever. But now we have an AlphaFold database where there is 200 million predictions. And since the field started to be open again, we coined that as an Alphafoldology because almost every other new tool coming after that somehow standed on top of the AlphaFold. And as my mentor, Janet Horton from the EBI said, that's basically usher a new era in biology, and that's where we are. So as I said, the Alphafoldology took quite early, and I would say that the first Alphafoldology tool was actually a Rosetta Fold, because Rosetta Fold was the tool that was able to reach to the AlphaFold 1 prediction accuracy from the 2018 cusp. And it's based on the David Baker lab in University of Washington, where they are specifically looking on protein design. But there were already in 2021, there was multiple different tools, and one of them is CollabFold, which basically democratized the access to the AlphaFold. Because you could use the CollabFold on Google Colab on basically any sequence you wish, and now there is multiple different CollabFolds for different models that you can use. And quickly it started to fill the proteins with the ligands. It started to be also for protein design itself, so you could basically, based on the structure, you could set up what the sequence should look like for that specific shape. And also the mutation started to feel, also there started to be quicker methods like ESM-fold using the protein language models. And then in 2024, it came AlphaFold 3 together with the companions, which started to able to generate with the generative AI models to generate also the ligands inside the proteins, which the term for that currently is co-folding methods. And last year we started to see also the fluctuations and dynamics models. So, where to get the models? Still the first place to go for the models is actually AlphaFold database at Alphafold EBIS UK. And there you can look for the proteins for almost any organism and look it up to see how the protein actually look like. And you get multiple different quality measures. So you get these colors actually says how confident is the model for that specific amino acid. So it basically says that these parts, this helix, he knows that it will be a helix. For this part, he will know that this is not only the helix, but also the folds around it. And also, it through the PAE, predicted confidence of contact, which is this matrix, he says whether these two amino acids are bind together well or not. So from this matrix, we basically can say that this part of the protein, which is here, this green square, is folded together well, and it will look like that. But here is the helix, and you can see that the helix together with the fold does not share any confidence, and therefore their relative or coordination and orientation can be basically almost anything. So we know that these two in comparison to each other should not be there. So when AlphaFold.db and DeepMind people looked into the human proteome, they found out that the resolved structures from the PDB are all really confident, while the unresolved are quite not confident at all. And basically that came up from the possibility that the protein can be unstructured. And as an unstructured protein, it can be so-called IDP, interestingly disordered protein, or the parts of the protein can be interestingly disordered. And for them, of course, there is no specific structure. And it looks like that the PLLDT is not only the quality metric, but it also gives the information about the disorder. And there were several studies to assess whether PLLDT is more the metrics for the quality or for the flexibility. And when we looked into the PLLDT in comparison to the molecular dynamics flexibility, RMSFs, it basically is quite correlated, and it's quite correlated also to the NMR flexibility, while the correlation to the X-ray B factor is really low. So it shows the flexibility of the protein, but it does not seem to be correlating well when you take into account the flexibility according to the interacting partners. So it is some kind of uncertainty, it is some kind of a flexibility, but depending on the method which you would like to compare. But the good thing is that it's comparable to what we see from the molecular dynamics simulations. Another thing which was trained later on by the based on AlphaFold was that you could take the AlphaFold structural confidence and compare it with, combine it with protein language modeling and also the variance from the ClinVar, and that gives the prediction of how much pathogenic your individual mutation can be. And this prediction basically was done by the team for all human proteins. So every amino acid, they've changed to every amino acid possible. So they've came up with the metrics of 20 to the length of the of individual proteins, and they put that data to Zenodo. And you can access these data for free at the AlphaFold database. So on the right side, when you have the structural view, then there is a possibility to color by the PLLDT or via the pathogenicity. And here is my favorite cytochrome P450-3A4, which is the major metabolizing enzyme. And you can see that there are some certain areas which seems to be more red, more pathogenic. So we tried to look into the structural information where these are actually located. And there is actually quite nice viewer from the Hege lab, from Tamás Hegedus. And he has a AlphaMycin data viewer where you can look into what mutations are actually available by typical mutations for that specific place. Because, of course, if you have a single nucleotide polymorphism, you cannot change any amino acids to any amino acid. Because that requires 3 nucleotides to change, so you can change complete codon. Yeah. So, if you have an amino acid and you have a single nucleotide polymorphism, you can change it only a subset of those 20 amino acids in general. And here, you basically can see which amino acids can be, the amino acid can be mutated to, and what will be the mean pathogenicity on that specific place. And since Tamás group is also looking for the membrane proteins, they've found out that quite often those pathogenic amino acids are actually in the transmembrane part of the protein. Like in this case, it is CTFR, cystic fibrosis receptor, and that one basically is a transmembrane protein, and this transmembrane helix bundle is the place of the most pathogenicity. We've looked into the channels because we are analyzing how molecules are interacting with the can reach the active site via the channels, as in our favorite cytochrome P450s. And we found out that while, of course, the most pathogenic part of the protein is the part which is surrounding the catalytic site, which is kind of obvious that if you made a mutation in the catalytic site, that you can kill the protein and therefore to have a pathogenic variant. We found out that if you go via some tunnels, and there is a whole methodology which tunnels can lead to and out of the active site, depending on the substrate and product. Those tunnels are actually slightly less pathogenic, but they are more pathogenic than the rest of the protein. So not only the active site itself, but also the paths which are leading to the active site are pathogenic as well. And we've seen the similar thing to the transporters, GLUT proteins, which are transmembrane transporters of glucose, and those are transmembrane, but also there is a pore where the glucose is passing by, and again, the transmembrane parts are more pathogenic, similarly as Tamas Hegerus seen, but the pore is more pathogenic than just the core of the transmembrane part. The AlphaFold2 database are just the proteins. So in order to put the ligands into it, you can go for the AlphaFill database, where they basically transplant from the known crystal structures, they transplant the ligand inside the structures of the AlphaFold, and you basically get some information about how good the transplantation is, and if it's okay with not much of the clashes, you can basically use that as a starting point for further studies. We also found out that it's possible to look for the channels in the AlphaFold structures. So we've enlarged our database of channels in proteins, channels DB, with the data coming from the AlphaFold database, because in the AlphaFold, because if we want to study the channel, we need to know from where to start. We need to have the annotation of the bindings of the active site. And quite often, the active site of the enzyme is filled with cofactor. So we can take the AlphaFill, we can take from the AlphaFill the position of the cofactor, and then run the mole analysis or K-Vra analysis to see whether there are some specific channels, and we can basically report them. And we stored that for the not only for the experimental PDB structures, but also for the AlphaFold. Another thing is that you can look, since you have a whole data for all protein structures, you can now do the proteome studies. And here is our try where we looked into the proteomes and how much secondary structures are in the proteins within the proteomes of individual organisms. And here, this is basically the organization. On the left side, there are the beta sheets, and on the right side, this is the alpha helix. And you can see that quite a lot of structures doesn't have any beta sheet. And if they does, then they have one or two or so. Basically, you cannot have one. In order to have a beta sheet, you have to have at least two pieces to form the beta sheet. And then more and more beta sheets you want to have, the less you find in a proteome. And on the side of the alpha helix, there is not many structures which don't have anything. And then there is several structures which have, let's say, up to 5. And then the trend is downwards again. or it is like some kind of power distribution or something like that. And we found out when this is the average distribution, but when we focused on different groups, we basically found out that there are three major differences. And the first difference is in plants, because in plants you have much more of those short proteins, the proteins which have only a limited amount of the secondary structures. So it might, since the plants typically have more proteins than the animals have, it might be that those additional proteins are usually the smaller ones, and maybe they are used as a signals from the plant to reach something else when they cannot move. Another thing which was quite fun to find is this nose on the rodents. And I'm speaking about the nose because when we looked into what is making this increase, we found out that this is the olfactory receptors, GPCRs. So they really, the film Ratatouille is based on real events, I would say, according to this, because the mouse really has, and the rats have better olfactory receptors than we do, although we still have the same thing, but much smaller. Okay? And what we are special in long proteins. But for this long proteins part, we think that this is actually an artifact stemming from the fact that the humans are more studied than any other organism. And therefore, the annotation of human proteome, where if you have really long protein, it can be connected to one, might not happen in the other organisms as well. So, speaking about the individual domains which are making the proteins, there is a nice study from the people behind CAT, from the UCL and Christine Orengo lab. And they found out when they analyzed the Alphafold database, they found quite a lot of new folds. But recently they've enlarged that study to add also the ESM fold atlas, which had 800 million proteins in it, and they found just one new fold. So based on that fact from increasing to the Alphafold database, we found substantial amount of the new folds, and now increasing even further by the ESM fold, we found just one. We can fairly say that we probably covered almost all fold space that is currently in the nature. But we can engineer new. And that's where I came to this part. Because as you've seen, I'm putting this small 3D printed statue about the 9AA, 9ADD as a gift for the speakers. And well, while the 9 is just a simple letter, A is actually DNA gyrase, so that's a real protein which has a hole required for deattaching the DNA. D for drug is, of course, the most important drug there is, which is coffin. But the letter D, I wasn't able to find any protein for the letter D. Because for some reason, you can have rounds in a structures. You can have long pillars, which are basically stiff. That's doable. But to combine these two things together in one structure, somehow the nature doesn't like. So for that reason, I've used one of the protein engineering tool, Chroma. I will be speaking about it yet. In order to generate the protein that would look like a letter D. So basically, I use the shape of the letter D, and I've asked the program to get me the sequence and to fold me a protein that would have this structure. And now we have the design of this lecture as a real design from the protein design. So the question is why we should want to do the protein design. And there is actually quite a lot of things which we can do with the protein design, and that's basically we can do the biosensors. We can set up, we can do the cancer therapies because we can basically make a binder for the protein which Genevri and to block it. We can make the vaccines also, like protein vaccines. We can do the drug delivery with the nanocages as the Baker lab is doing. So there is plenty of things where we can engineer the protein, and we can do also not only the protein switches, but one thing which is not here, we can design a new enzymes, for instance. So those are the tasks which are basically engineer tasks for which we would like to use the proteins, and we basically can do that because we now have tools to quickly generate the protein structures, like this ESM fold, which is really quick to generate you a structure based on the protein language model. Within a few seconds, basically, you get a new structure. And there are another tools which are basically reversed the point that you, from starting structure, you can basically design what sequence will fall the best for that specific structure. And that's the David Baker who basically established this field. And this is the chroma from their lab, which I've used to design the letter D on the statue. And there is a nice tool called Bindcraft, which designed the binding proteins, binding to something, to some target. And there are, again, the Rosetta Fold diffusion 2, which is quite similar approach again, which now can do the enzymes because you can design the reaction around how the reaction coordinates of the enzymes should look like according to the intermediate structures that would require the reaction. And the newest kit in this field is probably the Boltzgen, which is now quite popular version of Boltz 2, which allows you to generate new structures. And it was used actually also by quite a lot of people in this adaptive NIPAC competition, which was a competition trying to basically test whether we will be able to set up the infrastructure to react to a new problems, let's say virus X. And they've selected a NIPAC virus, which is a WHO priority pathogen, where there is no treatment for that one. It's quite deadly. And the major how it works is that it basically binds to ephrin receptor in human body, and that's the route how it goes inside. So the tasks for the competition was to find out the protein, find out the binders proteins that would bind towards the glycoprotein G spike protein and to block the interaction with the ephrin. So that was the competition setup, and people basically tried to generate the designs, and they've tried to use quite a lot of different tools to do that, mentioning the same which I've mentioned on the previous slides. And the adaptive has also increased the production of those proteins via the cell-free protein production, prototyping. So they were able to get to the expression level of those design protein to basically 90%. And the binding hit rate was 14%. So not every protein which was designed, out of those, I think I have a slide here. So out of those 10,000 candidates which were submitted, 1,000 was designed for validation, and out of them, 86% were expressed. So they had 881 expressed proteins. And with those ones, they had 99 confirmed binders. So basically 10% hit rate. And out of that, 26 design had actually a sub-nanomolar, single-digit nanomolar affinity. So they bound really strongly. And when we looked into the top binders, which came up, those results came up last week, and some of them even yesterday, for the top de novo, you can see that this is actually sub-nanomolar KD. So quite well, but not only but the problem here is that the protein, the new binder, has to be neutralizing one. So it has to neutralize the contact to the ephrin. That's the important thing, that the neutralization is basically the proof that it will work. And the second thing is safety check, because it can bind to everything. And in blood, we have a human serum albumin, which is the most typical protein in our blood. And therefore, if something would bind to human serum albumin, then of course it wouldn't work at all. And good thing is that no off-target binding to human serum albumin actually occurred with those top binders. And the first two top candidates were fully neutralizing antibodies. So, a huge success. But the third one and all de novo were not neutralizing at all. So simple binding assay, of course, is not enough. And the thing was that during the competition, the people were trying to optimize and game the score which was used for the computational design, and that was from Boltz to IP-SAE metric. But it seems that just to rely on one metric is not enough. So it seems that it still requires slightly more of the human thinking for now. Hopefully the robots will come just later. And the lesson here is that just to rely on one simple score is not enough. So the question for the last part will be where you can actually generate the models. Everywhere. As I said earlier, the Collapfold allows you to use those models in Jupyter notebooks, and you can basically run those Jupyter notebooks on Collapfold, or you can download and run them on your premises. It's up to you. Almost all the models are actually available on GitHub, so it's possible that you can download them and run them on your premises. There is a OpenFold, AlphaFold free implementation where the multiple companies basically collaborated together with the Al-Quraishi lab to set up the OpenFold, which is trained on also the pharmaceutical companies' data, but it's then provided open source and available. But you can also use the Alphafold server, because the Alphafold server allows you to do the quick trials and enables really nice graphical user interface where you can play just by having a Google account. You basically can play with the systems like this. So this is the cytochrome P450 together with its reductase, together with their cofactors, and together with the free fatty acids. So that's already starts to look like some kind of slightly complex systems. Here in Czech Republic, you have availability to use the A-infra resources on Foldify, which has multiple different protein folding tools, starting with AlphaFold 3, without the selection only for the selected ligands, but you can basically put any smiles in there. They have the AlphaFold 2, CollaFold, OmegaFold, and ESMFold. So you can use all of those, and the only thing you need is to have the life science AI to get there. But those tools are downloadable, so you can basically download them and run them on your computer. So one possibility is to use the Chimera X, where you have multiple options, and basically with the bolts, especially, you can download the bolts and run the bolts on your premises. It just requires a graphical card, a good one, because when I've tried to run it on my Asus computer laptop, I've tried to make a small 20 amino acid protein just as a test case, and I started during the evening, and I woke up in the morning, and there was still nothing. And then I've tried it here on my workstation, and it was done in a few seconds. So it really depends on the hardware. If you have a Mac, then there is also a finally method from the Apple, which is called SimpleFold, and the SimpleFold can be quite simply installed on Macs, and as my student Thierry found out, it's really quick and really simple to use. If you want to work with the workflows, then one of the workflows is, for instance, Galaxy workflow, which is used usually by the people who are trying to work with a large amount of data that have to be reproducibly dealt with. Like you do some sequencing, and then you do some quality assurance, and then do the models, and you can finish that with the protein models. And of course, we've heard yesterday, and during the tutorials, we will hear about Rowan. And in Rowan, there are actually several models for co-folding. And the Boltz 2 is able also to predict not only the confidence score, but also the affinity probability and predicted IC50. But a word of beware here, it's not, similarly as with docking, it's always necessary to validate those values, because when I tried to make a correlation between the values for those affinity probability and predicted PIC50, the correlation was really limited. I think it was something like 0.3 or something like that. You can also generate the ensembles nowadays. So there is, for instance, BioEmu, which enables you to make the ensemble of the structures. You can basically walk around, and that was trained on the MD ensembles. And here I have a few Alphafoldology hacks. The one thing is that the context really matters. Here is G quadruplex, which is the structure in the DNA, which is in the telomeres. And it is made by 4 guanines, which are basically making tetrates. And they should be on top of each other, and this is quite specific structure. And as you can see, the structure here resembles the tetrates, 2 tetrates on top of each other, but it's nothing really perfect. But when I've realized that actually there is a potassium ions in the middle, and I've added it, the model was immediately much more confident. So if you have some knowledge about something structural which can be added to the model, by addition to the model, you can actually help. And one of those things is actually lipids. Because if you add lipids, even with the AlphaFold free server, there are fatty acids. And with the fatty acids, you basically can make a model where this part will look like a membrane. And I've spent when it was my first study when I came here to Olomouc in 2010, and we published it in 2012. And we published the first model of cytochrome P450 on membranes, and it looked basically like that. So the AlphaFold 3 has it, even though he probably haven't seen my model. It's quite something, and that can be actually used, this trick can be actually used when you have 2 domains, which are extracellular and intracellular, like this FLT kinase, FLT3 kinase. And here you have the extracellular part and the intracellular kinase part. And from from the model, you would say that they are interacting to each other. But the PAE matrix gives you the clear warning that no, those are separate domains that are interacting only within themselves, but the interaction between both of those domains is basically non-existent. But the structure seems that there is a contact. Okay? So it's necessary to check for the PAE whether the contact is real or not, because of course the model is trying to make the model compact. But if you add the lipids, it immediately stabilizes the transmembrane helix, and if you add them well enough, it basically nicely gives you the extracellular and intracellular domains, and you get much more validity in both cases. If quite a lot of those tools, like ESM-Fold or the BioEmu, basically is able to do just monomers. But quite often we are interested in dimers. So if the method does not allow you to do that, one trick which can be used is to put here a linker. And if you would put there a linker which will be polyserine, which is quite polar and therefore it doesn't fold. It's quite often in the intracellular IDP parts of the proteins. You can basically make this link and fold the whole protein as one domain, and then just cut the linker afterwards. And you can actually do the co-folding with multiple ligands at once. So you can generate which ligands you would like to put. You can try with many of them. And then you can try to find out which one will actually bind to the binding sites with the most. And that can be some elite. Of course, it's not perfect, but it can make you elite what compounds or type of compounds will actually bind to that specific spot. But here I'm coming to the limitations of those models. And one limitation is the validity of the ligands. And that's coming from Wim de Haen's set. You can see here's plenty of nice looking molecules, really nice looking, right? And especially this one seems really nice, where you have this is the original smiles, how the molecule should look like. And this is how Boltzmann changed it. So you can see that it doesn't use this as an aromatic ring. And this one it made into an aromatic ring, and it changed triple bond to double bond. When I redone this with multiple different models, I found out that in chai, we basically got rid of 2 of those problems, but not with the triple bond. And Boltz 2 was okay in all cases. Yeah. So I'm hoping to see what are the other ligands going on soon. And now I have a new denaturation example. And that's coming from the same, this is the trip cage, which is a protein, mini protein, which was artificially developed to basically function as a folding example. Because you can fold this protein in a molecular dynamics simulation. But you can add the denaturation, and for denaturation you can add urea. And of course, when you add the urea, then you can lower the PLLDT, so the quality of the model, and therefore the flexibility should be increased. Right? Not the case. 5 ureas. You can see that the structure is rigid as ever. It's rigid as ever. It's still quite blue, and it's completely bruised this way. The lowering of the average PLDT is just coming from the fact that we are adding the ureas, which doesn't have any specified place, and therefore their PLDT is basically 0. And that, of course, lowers down the average PLDT. And if I would be just looking on a number, I would think that it can be used for denaturation analysis. But definitely it's not, because the protein is as rigid as ever, and that's definitely not what will happen in a real case. If you would run the simulation in urea, you will get completely flexible denaturated protein. Definitely not the protein that would be still as rigid as it was previously. So the proteins in the AlphaFold models and the Alphafoldology models currently are predicted as stable, and therefore their PLLDT would be unchanged. So to summarize, the AlphaFold 2 made a huge leap in the prediction accuracy that enables us to have a protein models together with the quality prediction. So we can see how good the model actually is, and therefore we can look on which parts we can trust and which don't. Without the open science and without the openness of the protein data bank, it wouldn't be possible, because the data in protein data bank are collected there for 60 years. Since those tools are openly available, there are more and more coming. And let's say every week or every month, there is a new tool and new tool and new tool enabling and pushing the boundaries of what we can do a little bit further. And there are many Alphafoldology tools that can have their use, and basically it's necessary to try them. And we will have to try them quickly, because the biology doesn't end with just one protein structure prediction, but that we predicted just how the native structure look like doesn't say that we know all of the biology of the rest, what will happen to the protein elsewhere. So yeah, thank you for your attention, and if you have any questions, please do ask.
And my question is about the stereochemistry problem you talked about. So I would assume that you have used potentials in bolts too, which is a huge help in the stereochemistry.
Yes.
And sometimes I think when I read the bolts 2 paper X, like when they introduced the potentials, they claim on some structures it kinda decreased the quality of the prediction. So if you have some advice when to use potentials and not. Okay, like it's nice to use them with ligands, but they also sometimes claim that it improves the structure prediction like overall, but sometimes it had decreased it, and I still couldn't like find in which cases of structures does using the potentials decrease the quality.
I must say that I don't have an answer to that, because I use the potentials since they are available, and they cost me nothing much more. And since they increase the quality of the ligands, I'm trying to use them as much as possible. But actually, I haven't tried to not use them and to compare side by side. Unfortunately, I cannot answer that one. But it might be a good question to ask in a Discord to Jonathan for his tutorial.
So certain proteins, dimerization depend on the concentration. So, and even mini proteins, like if you have tryptophan in between the proteins, they tend to stack. And so these protein prediction software does take concentration into account as well?
You can predict the dimers, trimers, tetramers, and look for the quality measurement, what will happen if you would add the additional well, let's say monomeric unit. And in my experience, when the protein tends to be dimer, for instance, then to make the dimer actually increase the quality of the protein. but if I add additional one to generally make trimer, it usually decreases the quality. So by that, I have some kind of a qualitative measure that would say to me, okay, 2 is enough, and I'll stay with that. So that's the way how to work with the concentration.
Thank you.
Okay, I see no further questions, because I suppose that you all want to have your coffee break. And before the coffee break, I have 2 announcements. One announcement is that please sign a new registration list, because we need specified dates when you will be here. And the second point is that by the end, after the Thierry Langer's lecture, we will have a photo of all together on the staircase. So hopefully the staircase will not collapse with us, because we've grown in numbers since the last year. And with that, thank you for the morning session, and enjoy coffee. So I see we are slowly reconvening after the coffee break. And in order to enjoy the coffee, now we will have the lecture about how to use not machine learning, as in most previous talks, but quantum mechanics in drug discovery by Martin Lepsik from IOCB. So the floor is yours.
Thank you, Karel, for the introduction, and also to you and Pavel for organizing this course and workshop. It's been a pleasure to be here with this type of lecture whose name has been the same for the years, but the contents has been really evolving, also including machine learning lately. And to show you how important the topic is, so there even are books on that topic, and this book, Quantum Mechanics in Drug Discovery, because this, and I will tell you the same for free, so you already saved a lot of money. No, I'm kidding. I will tell you something very much different what is in that book, because there are very many flavors of quantum mechanics and how it's used in drug design. But generally what I'll be speaking about is this binding of the ligands, small molecules to the proteins, and estimating the binding free energy, in other words, the dissociation constant. I work at IOCB in Prague. And I acknowledge first the team. So the core team, we are the 4 of us here, and the group now, which is a bit bigger, and many students who have bright capabilities. So the outline of the talk is the general setup where we are in what we want to find out, and how QM works in this area. The modeling is one part, but there is always experiment, which is the final truth, although with some errors. So it's important to have experimental data sets, well curated, and then I will show you how we extended SQM and used it in one case. So Karel already said yesterday that drug design and development is a lengthy process, which costs a lot, and it's also risky. So wherever you want to make it cheaper and increase your chances, you may want to use computations. And we are speaking today with this in this area, hit to lead or lead optimization, which means mostly small molecules and improving their affinities. There are two approaches generally: the structure-based drug design and ligand-based drug design. Mostly, we work in this structure-based drug design, which means we have a structure of the target protein, and there are processes of finding the ligand binding, which is traditional made by docking, but generally lately also with the generative AI. And also there are the ligand-based drug design, which will be more covered tomorrow. I will not speak about it too much, but generally what methods are used there. So for structure-based, you need to have the crystallographic or cryo-EM structure. Or again, generative AI generated, and either you do virtual screening through libraries, or you go and the engine there is docking and scoring. For ligand-based drug design, you want some informations about the ligands, which may be used in many areas, but importantly, you get to know the partial charges, for example, or some conformations. So there are different pathways where how you can predict the affinities. The classical way is to use standard scoring functions, which are very fast, but their accuracy is low. And there is not really a clear pathway how to make them better. In the recent times, there are machine learning methods that are fast generally, but always the question is how the training data are curated, whether they are accurate enough. And also you are bound to an application domain, so not so much predictions. If you want to go beyond that, it's very useful to use some physics-based models. Generally, there are those based on molecular mechanics, which goes to molecular dynamics, and then special methods like free energy perturbation, where you change one ligand into another one. The dream of alchemists has become true in the 20th century, so you can indeed change, at least in computer, some one entity into another and collect free energies. And such methods are really powerful and give you a chemical accuracy. So when you want to decide whether one ligand, like the new ligand, prospective ligand, would bind better or worse than your current one, you will use mostly in pharma industry FEP, free energy perturbation, and it will give you mostly the correct answer. The disadvantage there is that it is pretty slow. It runs several hours. It would run on GPUs. And also it's dependent on the force field accuracy. So if you come across a type of chemical problem that is not tackled well by force field, either for reasons of parameterization or for reasons of for other reasons, then you can run into problems. On the other hand, quantum mechanics could solve the problem. We have heard about DFT, which is kind of golden standard, but also faster methods. And DFT is mostly you would use some tens of CPUs. It could take days. So it's even slower than FEP+, but it's accurate and it has all the effects that force fields may not capture, like depending on the electrons, indeed, and whether it's applicable to biomolecules, yes, but they are big, so how to deal with that? Here are some examples how the quantum mechanics, what are the types of problems you can meet. So either there are some metalloprotein binding, there are some covalent binding, some electron densities which can be polarized, and obviously all non-covalent interactions like hydrogen bonding, etc. And everything is treated on the same methodological level, including the solvation. The other practical advantage is that, as opposed to force field, you don't need to parameterize any ligands. The only input is the total charge and the spin multiplicity. So that's a good thing. So why shouldn't we use this? And the answer there is the speed again. Looking at this graph, the scaling with the number of atoms is very steep for the very accurate coupled cluster method. So the error is only 1% with respect to the most accurate, but you can see the computational time in minutes, and here the logarithmic scales of number of atoms. So definitely this we cannot use. DFT is shown here, and then what could be used are SQM methods. So semi-empirics, okay, many atoms, thousands of them, but what about accuracy of semi-empirical methods? The accuracy is pretty low, as we can see here, of the native methods, like AM1, PM6 in here, and the spread is also big. What can be done with this? And here I want to acknowledge Professor Pavel Hobza, who's my previous tutor from IOCB, and since several decades he has been interested in high-level quantum mechanical methods, and as they could be only used in small molecules, he constructed such databases where you can see the prototypic prototypic like hydrogen bonding examples, and then some stacking interactions and aliphatic interactions. And these he calculated in the highest possible level. And such numbers and geometries serve later on to parameterize any SQM methods. So this is the way how to make the SQM methods more accurate. And this is my current boss, Jan Řezáč, who's continuing in this work by constructing so-called NCI Atlas with more than 20,000 data points, both equilibrium and non-equilibrium geometries. And as opposed to what Alexandre was saying in the morning, where there were reactions, so here it's really non-covalent interactions. So there is no bond forming, but very high accuracy of these calculations. And now hydrogen bonding, dispersion, all types of interactions are there, and the systematic development of corrections toward the semi-empirical methods could continue. And here we can see the results. Here I reiterate the original methods, AM1, PM6 with high average errors, and here are the new ones in red, where there are corrections for dispersion, hydrogen bonding, and sometimes also halogen bonding, X, and also chalcogen bonding and others. So a few types of mostly occurring methods. And you can see these errors have gone really down. So we have a method that is on one hand fast, and on the other hand it's reliable. So that's a great tool. But then all the binding occurs in water. So we need to do something about the water to treat all the molecules individually. It cannot be really done in terms of sampling, etc. So in this case, we use implicit models, which treat water as continuum. The original method was COSMO of implicit solvation, and you can see root mean square errors, which are here, and especially like COSMO and PCM are also original models, SMD, coupled with several semi-empirical methods. And so you can see the highest errors are there for cations especially. And what Jan Řezáč did in our lab is that he parameterized COSMO with some other corrections. So it's called now COSMO 2, and you can see how the errors went down. So now we have also the implicit solvent model, and putting these two guys together makes up a scoring function for us. So it's what you see as this is the binding process up there, and you can see individual terms which go like desolvation first of the protein and of the ligand, then changing of the conformations, then binding, and then resolvation. So it can be dismantled in our heads, but then it's put into these terms, but they are all added up. No weighting is there because they are all based on physical chemistry, and what we get there is SQM220 as a score, as a number. And this tells us about the affinity of individual ligands. We have used this in an SQM-MM setup where the ligand and its surroundings are treated at the QM level and optimized. The other are QM level frozen, and the rest is fixed. And we have worked on very many proteins over the years, but then we saw like with each of them we had different setup of the method, so we wanted to come up with a universal method. So that's where we had to find some experimental data sets where we knew, okay, this is what we want to achieve. But actually the experiment itself is not rock solid and one number, so we were looking into how the experiments, what their errors are. So you can see like between plus minus 2 kilocalories even you can get in some cases, but okay, it's mostly less, but you can see this graph. So generally R squared of 0.8 is what you can get as a Pearson correlation coefficient squared for two independent measurements from different labs on one system. So generally we want to make sure that the systems are reproducible. So we have collected a high quality data set where we took care about the experiment. First of all, we preferred KD or KI values over over IC50s because they are real thermodynamic ones. Then also we wanted to have the range of these values at least 2 orders of magnitude, because we wanted also not trying to hunt for some noise, but rather for real differences in affinities. And also we wanted structures, because we wanted to have the good results for good reasons. And structures, so mostly it's crystallography. And what you see usually is that you have like one crystal structure and then a series of ligands with measured activities. Here we wanted to have more control over the structure, so we really selected those systems that have multiple crystals, or for many of the complexes from the series. So you can see it here, like this number of crystals that we have are pretty immense. We have collected that from the literature, not to say that we have did it ourselves. But what we did ourselves was to prepare the complexes for calculation via QM, and that means we need to take care about alternative conformations of side chains, water molecules, hydrogens, tautomers, etc. Because all this makes a difference. And this is a data set called PLREX, Protein Ligand Reliable Experimental data set, and it's out there in GitHub. You can see it for use, whoever wants to test their scoring. It's freely available. Also, I must warn that it's a challenging data set, because it has things like big ligands which are flexible and have some higher deformation energies. There are metals, water molecules in the binding site, halogen bonding, deprotonation phenomena, and others. So it is difficult, yes. And we wanted to compare in the first step to the standard scoring functions. So we took from the CAS-F 2016 data set those upper scoring ones, and we went to compare them on the PLREX. And these are the results. We can see here in gray are the standard ones. So you can see glide, different versions, R squared of 0.4 roughly, and then it goes on and on and on. Then the next ones are some machine learning ones. Nowadays probably there would be others and possibly better, but these are from a few years, like two years ago. They didn't perform well, and here we have like our SQM score. So you remember the limit of 0.8, so we are roughly getting there, so that is a good sign. And we also compare to which is molecular mechanics amber, and then a hybrid of those two, which means geometries and SQM score calculated on them. You can see this, I have it once again here as importance of both the energies and the structures, because if we use good energies on intermediate structures or suboptimal structures, so you can see here that if you take it from from here, it deteriorates. If you come from here, it improves slightly. So this is the SQM220 method which we introduced 2 years ago, and now we went to like looking what is like the per target R squared, so performance, and we can see that we are pretty doing good across all the targets, whereas for example Amber does a bad job in some targets, but others manages. And comparison to DFT shows that we are on par, which is very good, but in comparison we are somewhere like 20 minutes on one CPU only. So we are roughly in terms of speed 1,000 times faster if we recalculate at one CPU. So we are roughly here in terms of accuracy versus and time. Okay. So PLREX success, very good, but okay, this was our system. So if we were not successful there, then we should probably end and do something else. So we wanted now to go to some real case benchmarks or data sets. So we compared to FEP Plus, and as I said, is the method mostly used in the pharma companies, and what they have is the data set of 8 targets where they have 10 to 40 ligands per each, but as I said, they mostly have like one crystal structure and the others are modeled, and this is their performance, R squared of 0.56. So when we and also like what we had in PLREX are ligands that are quite dissimilar. Some of them are similar, some of them are dissimilar. Whereas here, because of the construction of the alchemical mutations, these are so-called congeneric series, so very similar one to another. FEP Plus had 0.6. We applied SQM220 scoring and we got 0.38. So no, not really great. And we could see that in 4 of the targets we didn't have any correlation. So we looked into this, why that was, and found out that there were really big troubles in the starting structures, which we normally optimize, but the optimization goes only to the first local minimum and may not resolve all the troubles there. Whereas they, in FEP Plus, they run molecular dynamics, so it has more time and also energy to overcome some barriers and get out of the problems. So what we did with a very simple problems, just to see what it does, we corrected manually some positions of hydrogens, etc. And lo and behold, 3 of the targets got corrected, and we got quite close. Still worse than FEP Plus, but quite close. So that is good. We did also the same with docking, so it also made a similar thing as the manual correction. So again, this shows how reliable we are on good structures on the input. So this shows us that we need something on the input which needs to give us the good structures. So traditionally it's docking, so we tried docking first, then more modern is generative AI, so co-folding methods come as the next. So what about docking? We can dock our structures, take the best pose, and do the scoring. So you can see here it's this standard docking, and this R squared drops dramatically. When we take all 50 poses, calculate all of them with SQM scoring, and let SQM decide what is the best and not the docking program, it improves a bit. But we want to go further. We analyzed this and saw, okay, even 50 poses may not have in all times the correct pose in there. Correct, I mean near native. So that's why we made exhaustive docking, which then generates thousands of structures and like multiple runs, etc. We can again take best pose. It improves, and this is because the good pose is there already, and we have a series of steps to improve it. So that is the way to go, but this is at the cost of so many computations of thousands of poses by SQM. So it becomes so many hours, yeah, like 60, 70 hours per ligand. So it's not really economical in this sense. We wanted to see also, first of all, how the native pose is identified in docking and in SQM, how it can like decide what is the good one, good pose. So for this we made a study where we had a few proteins, and we saw the energy of the poses with respect to RMSD. We always knew the crystal structure, and this is how it should look like between 0 and 2 angstrom RMSD of the poses. It should be zero-ish energy, relative energy, and then all the other poses should be worse. The green and red are SQM and amber, and they both show slightly worse energies for already higher RMSDs. So that distinguishes the good and bad poses. Here it's a problematic case where you can see in between 0 and 2 there are energies which are better than the crystal. So this is a problem because it means these are false positives. So there is some problem, and this study was done on rigid structure, so it can be like this. But very generally, if we look in these structures and look like these high columns are number of false positives, and they are from standard scoring functions, and these red columns are false positives from SQM 220. So we can, or SQM can recognize the native pose. This is good. And the next step is to use AlphaFold 3 on PLREX. So this again should be a very simple test, because PLREX was done as a mostly structures from the PDB, so it should all have been in the training set of AlphaFold. But still, we could find some issues. So first of all, you can see the poses mostly fall into this 0 to 2 RMSD angstrom, for the ligand, so that's very good, but then the tail goes up to 8 angstroms. So what is happening in the problematic cases? We can see it in this colored figure, where there are poses, like green is the crystal and the colored are the poses from AlphaFold. We generate multiple poses. And what you can see here is one like S stereoisomer and R stereoisomer on this methyl. And what you can see here is that AlphaFold 3 not only generates R and S, but also generates different conformations. So this is to show that you can have the correct structure in there, but you can also have like other structures, but also other stereoisomers. So it's important, first of all, to make a sanity check that the output structure is the same as the input in terms of the stereochemistry, and second, to compare the towards the crystal structure. Also, you can we saw some errors which it learned from the PDB, and it was consistently giving them. So, for example, here you can see the hydroxyl group of the ligand, which is shown consistently in this position. You can see here it's shown in the PDB 2D, and there was, like in the series, there was one ligand which had this in the PDB. So already you start thinking, okay, so this guy is somewhat troublesome, and this gave like this one. And the trouble was, you can see the hydroxyl in here, and the trouble was that all this which went into the PDB was wrong. When we compared back to the literature, that like between the literature and the PDB, they swapped the stereochemistry, and then they solved the crystal structure. Obviously, this tiny change will not be seen in the electron density, so they consistently put it wrong, and AlphaFold gives it wrong. And finally, we made it originally with this, and the scoring was fair but not great. But then when we spotted the error, we corrected the structures from AlphaFold, and the scoring, SQM scoring, got a much better correlation with the experiment. So it's always good not only to check PDBs, but also the chemist's information from the paper. Another case was here, where you can see the electron density. The ligand is from the crystal is in green, from the AlphaFold 3 is in blue, and we can, when we go down to this area, you can see like it looks like AlphaFold 3 pose fits better to the electron density, this blue thing, as opposed to the crystal, which goes kind of to this red region. So I checked first of all the PDB. It showed the structure, R and S enantiomers here, one R to S enantiomer. So again, I went to the literature, and this showed that this structure should indeed be cis-racemate. So in other words, like these two positions are on the same side. And lo and behold, you can see they picked only one cis-racemate possible, RS, and it can be also SR. And what AlphaFold picked was this SR. So so they should at least put both, or if the other would fit less, which is the case, they should put really something what AlphaFold shows. So that's probably what out of the racemate, the crystal took only one. So it can be very useful to see the results of AlphaFold, which can point you to some errors in or omissions in the starting structures. So very generally, when we did SQM scoring on AlphaFold 3 structures, we saw compared to the crystals, which are in blue. Let's go to the average. So the performance drops for docked or different sorts of AlphaFold 3 scored, but it doesn't do a very bad thing in many cases, like here, yeah. But yeah, somewhere there are troubles, but generally it looks it could be very much useful. Sometimes we also found AlphaFold 3 poses that were better than reality, and that for 2 reasons. First of all, it gave better energies than reality, than it was in the crystal structure, and second, also better structures that we could see by like the type of non-covalent interactions, etc. So there we are not really sure what's happening. Maybe we are missing something in the model, and we are still investigating this. And now the big test, obviously, is to see the systems which are outside of the training set. So, and those are very difficult to find out because it must be some very new data, like freshly published, etc., or unpublished. And we could see in some cases there for these 10 other or a few targets, we could see like good average distance for the ligands. In 3 of them, there is something fishy, so AlphaFold 3 cannot predict the structures at all. And for the scoring, it looks like similar to the building procedure, but it's automatized and it's faster. So this is what we are investigating right now. And also we heard about Boltz 2x, so we tried it and tried its prediction of affinities as well. And what we generally found on PLREX is that the weak ligands are pretty dispersed in terms of like this is the predicted versus experimental, whereas the strong ligands are underestimated with respect to the experiment. Okay, and now just a brief jump into like where this can be used. It can be used not only in the small molecules, but also in quite big ligands like insulin, and obviously the insulin receptor is huge. So here it's the 51 amino acid long insulin, and it's like 2 chains, A chain, B chain, and these analogs are used as for insulin for the treatment of diabetes. And you can see it's like the small thing here in green and black, whereas this whole thing around it is its receptor. It's very flexible. and thus also its resolution is very low, 3.8 angstrom. So this is a case where other types of modeling need to come into play, and that is mostly molecular dynamics, and how to make this molecular dynamics and SQM work. It was mostly the project of Yevgen Yurenko, who is here in the audience. So this is just to see what are the conformations of the insulin receptor. So this needs to be taken somewhat into account. And here already are results like on the interface. You can see there are like different conformations of the protein and of the insulin itself, and also some like type of analysis over 3 trajectories of molecular dynamics of how hydrogen bonding, what is important and what is not. And all these statistically were turned into like snapshots, and the contributions of individual residues were assessed by a so-called virtual glycine scan, which means that the first with SQM 220 we calculated the interaction of the residue in question with insulin, and then it was mutated to glycine and calculated again, and the difference means like how important this residue for the binding is. And you can see for several residues here of the insulin, so how important they are. So you can see they can their contributions can go up to 30 kilocalorie per mole, and all these colors are different protocols. So you can see how much they can change, and also you can see the standard deviations that's from different trajectories and different snapshots. So this means it's very important to know the limitations also of the flexibility, etc. But generally the trends with respect to the experimentally known importance of the amino acids were captured. And also, like, why this B13 has so huge numbers. So that is because it's a type of interaction. You can see B13 up here, which may make some highly charged cluster. You can see two plus charged arginines, 1 glutamate, and these are notoriously troublesome to describe both in quantum mechanics and in molecular mechanics. In this case, this was we studied it with classical molecular dynamics, and we could see that a classical Amber force field, for example, FF19SB, overbinds. In other words, like shows something like 4 kilocalories per mole for this type of salt bridge, whereas CHARM has it like 2 kilocalories per mole, and ECC, like this charge scaling approach, makes it 1, and the reality should be somewhere around 2. So this is to show that not everything is solved. You can still work on something and discover new things, new approaches, new methodologies. So I would just sum it up here that we have developed on one branch of quantum mechanics, like on the semi-empirical methods, over the years or decades, maybe even a method for scoring which can give reliable predictions of affinity, of the accuracy of DFT, but at 1 CPU in 20 minutes. The structures are of high quality, and it can get some insights into the design of other ligands. It was tested on diverse set of curated data. We are testing further and developing further. The PLRX dataset is available, so if you want to test, you think you have a great scoring function that can tackle anything, go to PLRX and try it out, whether as a difficult test. And we have the software which we license commercially to pharmaceutical companies, but also we are thinking about academic licensing. And it is obviously superior to quick approaches like MM and comparable to FEP+. And the new developments in the AI-generated structures show that it can be used as a good input for SQM scoring. As opposed to docking, it has also the protein moves. So docking only can give you at best some flexible amino acid, but so I would say do explore there, but be careful for some errors like this switching of stereochemistry. So you need to then go back for some physics-based method again. So that is all. Thank you for your attention, and if you have any questions, I'll be happy to take them. Yeah, that's a great question. So there are two reasons. One reason is the real errors and lack of, let's say, care in preparing the structures of the MD people. I don't blame them because for them it's enough. Yeah, for force field plus dynamics, they will improve them and over the course of MD. So that's good. But for us, we would have to prepare them ourselves in terms of like what we did with docking. So we could. I would say 0.47 is pretty good as we got with docking. So that's one source of troubles, the input structures, and the other indeed is dynamics, where like what you see here, even when we do very accurate single point binding, so when we do multiple conformations, and so then the standard deviations can be plus minus 7 kilocalories. So there obviously we may not be able to capture like the correct behavior, let's say. And maybe there is only as well as a third aspect, and this is that some systems are really not amenable to scoring or SQM scoring, and the reason is that they may be entropy driven, for example. Yeah, so or there may be some other factors that it's really difficult to, or some like water molecule binding. We have water molecules also explicitly in there on top of implicit, but sometimes like to capture this quantitatively, like some water molecules expelled from the binding site, etc., it's difficult to describe. So there are several other possibilities.
covered as well. So yeah, we can close this session. Thank Martin again.
Thank you.
And we will meet in 1 hour after the lunch break. Have a good lunch.
Okay, so welcome back for the afternoon session. And we'll start with Peter Ertl from Ertl Molecular, formerly Novartis, who will be speaking about substructure analysis in drug discovery. So the floor is yours.
Okay, thank you, Karel. Thank you for invitation and good afternoon, those who came. Yeah. So I came myself from Basel in Switzerland, when I served for more than 20 years as a director of chem informatics in Novartis Pharmaceuticals. And last year I left Novartis, and now I am simply enjoying myself by amusing with chem informatics research. As you can see there, I will speak about substructure analysis in drug discovery. I will speak about rings, scaffolds, functional groups, and substituents. I will start by these 4 plots. I suppose most of you, practically all of you sitting in this audience, are dealing either with molecules or proteins or bioactivity data. And even if you are not doing this many years, maybe 2 years or 3 years or so, somebody of you, I suppose you notice already one fact. And this fact is that the amount of data we have to process in our daily work is growing very, very fast. And this is displayed on these 4 graphs, showing growth in size of 4 databases that are used basically daily in drug discovery business. This is the database of chemical abstracts. They claim this database contains all molecules published and patented, currently some 270 million molecules. Then it is the Campbell database containing bioactivity data and molecular data from medicinal chemistry journals. CCDC, that is Cambridge Crystallographic Database, containing X-ray 3D structures of small molecules. And finally, Protein Data Bank, containing 3D structures of proteins and protein ligand complexes. And you can see this by yourself. The shape of these 4 curves is very similar. It is nearly identical. And that's basically exponential growth. I am doing these statistics nearly more than 10 years. And every year I wonder when this exponential growth will stop. But apparently there is really no stop of this exponential growth. These data are from December last year, December 25. So these are very actual data. And these are existing synthesized molecules. I am not even touching these virtual databases, which can be very readily synthesized. Enamine came with this when they provided a real database. Real stands for readily available. These are molecules which may be easily synthesized for that they have the synthetic protocols. And then other compound providers followed suit. This database, size of these databases is billions. Recently I have seen even trillions of molecules. So this is incredible. But for the topic of my presentation, I will stay with the word or space of existing molecules. It is large enough. This large number, this is something like double-edged sword. On one side, those of you tried just downloading, storing in the computer on the disk is quite challenging, and processing of these hundreds of millions of molecules. So this is quite tricky. But on the other side, this large amount of data, this is very good because this provides a lot of training data that can be used for development of models for example, for prediction of biological activity. And I will show you some examples of these models. Most of my analysis is based on the Campbell database. I suppose most of you know Campbell, are familiar with the Campbell database. Those of you who are not, I strongly recommend to become familiar, because Campbell is really indispensable resource for both theoretical chemist, chem informatician, as well as experimental medicinal chemist. This database is maintained by European Bioinformatics Institute in Kingston. They are simply collecting data from medicinal chemistry journals, about 20, 30 journals, and they went back 40 years. Last release, Campbell 36, contained nearly 3 million molecules, 24 million bioactivity data points, and nearly 18,000 different targets. In my analysis, I will use a method that is called substructure analysis. Unlike standard molecular processing, when we are analyzing and processing the complete whole molecules, in substructure analysis, we are simply focusing to some specific parts of the molecules. We can see some examples here. Yeah? These may be scaffolds, this orange part, rings, blue part connected by linkers, red, or these may be substituents or functional groups. I will try to cover some of these substructures. And I will illustrate some of my results using this set of free cheminformatics web-based tools which are available on my website. After leaving Novartis, I am simply amusing by development of these web tools for medicinal chemists. As I told you, these are free. Everybody can play with them, but please not now, because if 50 people are starting to play, they will crash my server, and I want to I want to show you some live examples on this. So I told you I will speak about substructures. I will speak about these 4 substructures. I will speak about rings, scaffolds, functional groups, and substituents. So let's start with the rings. Here we can see some examples of rings from bioactive molecules. I will use the terms ring and ring system more or less interchangeably. Basically, this is a system consisting of either 1 ring, or 2 rings, or 3 rings, or 4 rings, merged either by fusion, when the 2 rings have 1 or 2 or several bonds together, or the spiral, when the 2 rings have 1 atom common. You can see some examples here. Rings do not contain any non-ring atoms, with exception of the atoms connected by double bond to ring system. These are mostly oxygen, but maybe also nitrogen, carbon, and so on. We are keeping them because they really are very important for the ring properties. If we would cut away this carbonyl oxygen, we would destroy aromaticity, for example, which is extremely important for ring properties. So why to focus on rings? Rings are clearly the most important part of bioactive molecules. They give molecules their shape, determine whether the molecule is rigid or flexible. They keep substituents in their proper 3D positions. They influence global molecular properties, hydrophobicity, polarity. And in many cases, ring atoms are also directly involved in the interactions with the proteins. The kinase inhibitors are typical examples. No wonder that more than 99% of bioactive molecules contain at least 1 ring. In pharma industry, we do not like these spaghetti-like molecules without rings because they may be present in many conformations, and it is difficult to fix conformation that is required for optimal binding with the target protein. And rings play also important role in many of these classical standard medicinal chemistry techniques, like scaffold hopping, combinatorial chemistry design, DEL libraries, these are DNA encoded libraries, patent analysis, and so on and so forth. So here we can see the most common rings extracted from bioactive molecules. These are benzene rings, pyridine, piperidine, piperazine, pyrazine, pyrimidine, and so on. Just one short remark. If some of you hope to find a job in pharmaceutical industry in the future after finishing your study or PhD or whatever, or postdoc, and you are not familiar with these names I just told you, then learn these names, because first of all, you will get the good points by interview, and second, anyway, you will need these names in the discussion with chemists. So these are the most common rings from bioactive molecules, from Campbell. Under bioactive, I understand any molecule with activity better than 10 micromolar or basically any target. But there is nothing really special about these most common rings. Benzene rings would be on the position 1 in any large molecular database, bioactive, common, pesticides, even toxic molecules, because it is very common ring. And also these other rings are simply common, and they are at the top. So what we are actually interested are rings not absolutely most common, but relatively most common, which are much more frequent in bioactive molecules than in common molecules. For that, of course, we need some reference set of rings present in common molecules. And for that, I took rings from another large database, Zinc database. This is public database containing molecules which may be purchased, which are available for purchasing on the shelf. Currently, I used set with about 12 million molecules. So in this green square, what we have on the previous slide, rings from bioactive molecules, on the right, rings from the common molecules. Yeah, phenyl is always on the top, as I told you before. We can see, but the pictures are similar. Yeah, we can see some short, small differences. Yeah, that pyridine is here the second, here is the fourth, and so on. But it would be pretty boring and complicated just to compare two images. But there are better ways. This is already more useful, more interesting. On this graph, we can see 80 most common rings from the bioactive molecules and 80 from these common molecules. Altogether, I think 150 unique rings. The x-axis, this is the ring frequency. On the left, we have the very common, very frequent rings. On the right, well, also common, but maybe slightly less common. But what is interesting, this is this epsilon axis, yeah, this one. This is the ratio, actually logarithm of the ratio between the frequency of the rings in bioactive set and in common set. So this means in this green area, these are rings which are much more frequently present in bioactive molecules, and in red area, rings which are more frequently present in common molecules. And we can already see some interesting facts from this graph. In this green set, what we are mostly interested in, we can see fused system 6,6, 6,5 with several nitrogens. In this inactive set, we have aliphatic rings, spiral, a little bit exotic system. They are pretty common in building blocks, but apparently these rings are not so common or frequent in bioactive molecules. They do not bring bioactivity in the molecules. So one can use this, for example, for virtual screening, or when we are enhancing compound archive, when we want to purchase molecules for screening. We will focus on molecules in this containing rings from this green area, and we will try to avoid molecules containing these non-interesting rings. But this is still far from optimal, because this is static graph. This is static graph with about 150 rings. And what we ideally need is an interactive tool able to display 10,000 or maybe 50,000 rings that would be interactive. The chemists simply move the mouse and watch these rings. But also this is possible, and I will show you this. But first of all, something how to generate such a graph. So first of all, we somehow need to characterize our rings by calculated properties, so they may be processed in the computer. There are many different descriptors one can use to characterize molecules. Fingerprints, common fingerprints, that's very common descriptors. I suppose many of you used these descriptors. But in this case, I used descriptors developed specifically for characterization of rings and scaffolds, and they are called scaffold keys. Basically, this is a set of 42 simple topological descriptors that provide the best recall of bioisostatic rings. That means rings that are present in molecules active on the same target. These 42 simple descriptors characterize, well, you can see this here, number of atoms and bonds of certain types, spiral atoms, exocyclic atoms, number of nitrogens, oxygens, and so on and so forth. and the properties of these smaller rings forming the whole ring system, whether they are aliphatic, aromatic, size of these rings, and so on. As I told you, these descriptors have been selected from the larger number of possible descriptors to provide the best recall of bioisosteric rings. And you can see example on the right side of these graphs. Of course, this may be used also for ring similarity searches. Rings are similar when they have similar scaffold keys. This yellow systems with yellow background, these are queries, and we can see set of similar rings. This blue one, they are really known to be bioisosteric, present in molecules with the same type of biological activity. This white, well, this doesn't mean that they are not bioisosteric, but they have simply not been tested, or they are not, bioactivity data are not known. But you can see that they are very similar, and I suppose if they would be tested, most of them would be shown to be bioisosteric. So now we have about 40,000 common rings extracted from the Campbell database and 42 descriptors for these rings. So we have a huge matrix, 42 by 40. And we need to squeeze this multi-dimensional matrix into 2 dimensions to be simply to show the diversity, to show the data on the computer. One can use many methods. You know, you are familiar with this modern method, UMAP and TISNE or whatever. For this case, I used principal component analysis. This is a relatively old-fashioned method, but it has clear advantage. The axes have physical meaning. They are simply combination of the different descriptors. So in this case, we can see this 2-dimensional matrix. On the x-axis, this is basically properties related to the size. On the left, smaller. On the right, a little bit larger. This y-axis, this is something like feature richness, or it may be complexity. How many different structural features this ring contains? Number of heteroatoms, number of, spiral system, and so on and so forth. At the bottom, these are systems with low complexity. At the top, system with high complexity. And we can already see, I want to say this, the color points. Color points, these are rings present consistently in bioactive molecules, at least 10 different bioactive molecules, and they are color-coded based on the target. We have seen before in Campbell there are some 18,000 different targets. I classify them into basic main categories. So here we have GPCR, kinases, proteases, other enzyme, nuclear receptor, ion channels, and so on and so forth. So we can see that this bioactivity is not distributed randomly. It is distributed in several clusters. And what is also interesting, there is such a diagonal structure of this colored point. Remember this, I will show you this in a moment. And we can also see that the bioactivity is mostly concentrated in this, somehow in the central area of this graph. So some certain level of complexity is necessary. Rings which are which have very little complexity, like here, or very high complexity, they do not contain many bioactive molecules. So we need some certain level of complexity. And we can also see that the color points with the same color, green, blue, or so, are somehow clustered together. And this is understandable because, of course, similar rings will show the similar type of biological activity. So now let's try for some demo. So this is my website with these different tools I have been telling you about. So let's pick this tool, which is called Magic Rings. So this is what you have seen before, but this is interactive. We can move the mouse and see the rings. On the left, system with 1 ring. Now we are in the area with 2 rings. This is this diagonal structure. This very dense area, here we have system with 3 rings. And on the right, we have a large system with 4 on 5 rings. When we analyze this interactively and identify area which is of interest, we can simply zoom at it, yeah. So this is this small zoomed area. We have here many magenta points. These are rings which are present in molecules active as GPCR ligands. So apparently this part of chemical space is active or focused on GPCR activity. So these are these reference molecules, and now we can simply move the mouse and see the rings which are in this area. And if we find something interesting, we can click and select these rings. Yes, some of them are not so similar. This is a result of the squeezing of these 42 dimensions into 2 dimensions. Therefore, some manual selection is necessary. And once we identify those interesting rings, we can move to the left to this arrow, download selected rings, reference, as well as those we have selected as smiles, and do whatever we want with them. Order molecules, screen them, and hope for the active molecules. This is a visualization of all molecules present in this particular area of chemical space. So I have told you before about these small clusters of the molecules of the rings of the same colors. Here we can see, for examples, GPCR ligands, kinase inhibitors, protease inhibitors, enzyme inhibitors. These color-coded, there are really many molecules with the same type of activity present, white, not tested. But I suppose you see they are pretty similar, and I suppose if they would be tested, they would also show GPCR activity or kinase activity. So now we are moving from rings to second substructure area, to the scaffolds. Scaffold is simply part of the molecule when we cut away all non-ring substituents. So basically scaffold, this is 1 or 2 ring system connected by linkers. Here there are some examples. At the top row, these are molecules. At the bottom, these are scaffolds. In this case, this scaffold consists of 3 rings and 1 linker, carbonyl group. Here we have 1 linker, just 1 carbon atom. What I told you about rings is even more true about scaffolds, because scaffold is really the core of the molecule. So the influence of the 3D structure of molecule, flexibility, whatever, aromaticity, flatness, influence on global molecular properties, that's exactly the same also for the scaffolds. Here we can see some common scaffolds from bioactive molecules. Phenyl is always on the top, but in this case, these are molecules containing just 1 single ring. So there are how many? 12,000 different molecules in Campbell that they are just simple phenyl derivatives. And we can see here also some larger rings, larger scaffolds, for example, particularly this one. These are bases of many bioactive molecules. Yeah? We can see adenosine scaffold, which is basis of 12 approved drugs, flavone and coumarin, which are based on many natural products. Then also this scaffold, basis of many oncology, which one? This one. Many of antiviral oncology drugs, and this particularly large scaffold, this is present in how many times? Nearly 400 times in the Campbell. This is basis of many peptide antibiotics. Scaffolds are basis of one very important technique in drug discovery and medicinal chemistry, and this is bioisosteric design. If you would be lucky and find a job in pharma companies, you will be doing this nearly daily, yeah? Bioisosteric design. Bioisosteric design is simply a replacement of a substructure or functionality in bioactive molecules. This may be rings, scaffold, functional group, substituents, but most commonly this is the scaffold. So this is replacement of a functionality having similar size and physicochemical properties. Bioisosteric transformation are used either to keep or even improve the bioactivity of the parent molecules, but optimize other properties, selectivity, transport properties, permeability, or remove side effects, remove toxicity, or design molecules which are easy to synthesize, or simply to avoid patented structural features. There are many tools for bioisostatic design, commercial, but one can use also this tool available on my website. One can simply draw scaffolds and get a list of similar scaffolds. Similarity is calculated by these scaffold keys, and the results are displayed on this graph. There are two ways to color these similar scaffolds, either based on the number of molecules having this scaffold in the Campbell database. The green are most common, white, they are present only one. So this, how many times they are present, this tells us something about synthetic feasibility. If the scaffold is common, it is apparently easy to synthesize. If it is rare, it is difficult to synthesize. And there are another way to color identified scaffold, and this is this target properties. You remember these 10 different target classes, which I have told you before. So now simply the scaffolds are present on the preferred target of molecules containing these particular scaffolds. In gray, there is the promiscuity, there is the mixture of molecules having this scaffold. So let's try this interactively. I will see how many people are playing with these web tools. So let's try to draw some simple scaffold. We can which display we want based on frequency, based on. So now it is searching. The smiles has been sent to the database, and now it's doing similarity search. Okay, good. So it came. So we would probably. These are green, these are most common. So this can serve as some inspiration for the design of bioisostatic scaffolds. So probably we would start with those which are green, so with this one, this one, this one, this one, either to synthesize them or to identify molecules having these scaffolds and purchase them. So now we will try, now we will try to make similarity based on the target class. As I told you, in the background, there is a big database of more than 40,000 scaffolds characterized by these different 42 scaffold keys, and the similarity is calculated as the similarity in these scaffold keys. Yes, so green, these are kinases, violet, GPCR, yellow, these are scaffold active on ion channels, and I even forget what are these different colors. Good, so now we are moving to the third substructure, functional groups. Those of you who made a course of organic chemistry, I suppose know very well what is the functional groups, because the learning about acid, amines, aldehydes, chlorides, and so on form the basis of chemistry curricula. But those of you who didn't make the course of organic chemistry, basically functional groups is a group of atomous bonds that give molecules its characteristic or is responsible for the characteristic chemical reaction of the molecules. So functional groups are therefore really a basis or cornerstone of synthetic chemistry, medicinal chemistry, pharmacokinetics, toxicity, spectroscopy, and also chemical nomenclature. So therefore, cheminformatics study of functional groups can be of good importance. Actually, I came in closer contact with functional groups in Novartis a couple of years ago, when colleagues from the natural product unit asked me to identify functional groups in natural products. They simply wanted to make derivatization of natural products and asked me to simply identify these functional groups. Normally, one identifies functional groups by set of substructure searches. One is to have a set of substructure queries, smarts, and then runs these substructure queries and identify functional groups in these molecules. But in this case, I completely failed. And the reason is that structure of natural products are very, very complex. Do you know what are natural products? Basically, very briefly, natural products are molecules produced by living organisms. Molecules like antibiotics produced by bacteria, or alkaloids produced by medicinal plants, or various toxins produced by these snails or tropical forest frogs and spiders and so on and so forth. The organisms are using these natural products basically as a chemical warfare, either to protect themselves from predators or to be used as a means of attack, like these different toxins. And that's the reason why natural products have been optimized in very, very long, 2 billion years long natural selection process for optimal interactions with biological molecules, with proteins. Natural products are a very useful source of bioactive structures in drug discovery, but that also structures of natural products are very complex. And therefore I fail with my substructure searches. So I had to develop a completely novel method to identify function groups based not on a set of substructures, but based on this iterative algorithm. Here we can see a very, very brief outline of these algorithms. We are looping through all atoms in the molecules. One heteroatom is identified because function groups are related to heteroatoms. One heteroatom is identified. It serves as a seed, as a starting point of a novel function groups. All other heteroatoms connected are added, and also carbons connected by double bond. And when we cannot add any new atoms, so this is basically function groups. There are many, many other fine tuning, but I do not have time to go into details. So basically here we can see some examples of functional groups for these three molecules. There are very simple one atomic, like this amino group or phenol group, classical one, like this amide or carboxylic acid, but also larger conjugated functional groups. If somebody is interested in details, he or she can find details here. And there are two open source implementation available, one in Java, one in Python using Ertl kit. I didn't do this myself. Yeah? This has been done by colleagues using this algorithm described in this paper. And here are the most common functional groups. Amide, this is from PubChem. PubChem is another very large open chemical database, about 120 million molecules. Campbell, you already know. Amide is always the most common, then ether, and tertiary amine. And then a little bit, we can see a little bit differences. What I have shown you before, this has been tip of the iceberg. Most common functional groups, but there are not many. About 60 functional groups are common. But you know, distribution of any substructure feature, functional groups, rings, scaffolds, this is a so-called Taylor distribution or a long tail distribution. We have a very few common and large, long tail of very rare functional groups or substructures. So here I selected some of these very rare functional groups. If somebody feels himself or herself expert in chemical nomenclature, then one should try to name molecules. This would be pretty complicated. And this also nicely illustrates the fact what I told you before. It is not possible to base method of identification of substructure groups based on just a list of predefined substructure. Because for that we would really need millions of substructure queries. So therefore it is really necessary to have a system based on this algorithmic loop through atoms approach. Okay, and then this is last slide concerning the functional groups. Here is a comparison of distribution of functional groups in 5 large collection of molecules. Here you have drugs, bioactive molecules, patent, synthetic molecules, and natural products. This is the relative distribution. And this is interesting. In some cases, the distribution is very similar, like here, or I don't know, here. But in many cases, we can see really strict differences. And the largest difference is that this green bar, this in this case, this case, this case, is completely different than the bars of other colors. And the reason is, you know this already, I told you before, these first 4 databases, drug, bioactive molecules, patent, synthetic molecules, these are mostly molecules synthesized by humans, by person. By these natural products, these are molecules synthesized by complex enzymatic reactions in animals. And apparently, these reactions by humans and reactions by these enzymes and animals are going completely different way. Good, and we are coming slowly to the last substructure features. I'm going to speak about, and these are substituents. Substituents, this is simply part of the molecule connected by a single bond to the rest of the molecule, usually connected to some rings, either aliphatic or aromatic ring. Here we can see some examples of common substituents, again from these bioactive molecules in Campbell. The concept of substituents and their influence on molecular properties is actually one of the pillars of the organic chemistry and physical organic chemistry, and also QSAR analysis. I listed here some important milestones. Yeah? Hammett contribution. If you took part on the course of physical organic chemistry, I suppose you are familiar with this. If not, well, Hammett's contributions about the effect of substituents on reactivity. Hammett introduced so-called Hammett sigma constants. Basically, this is a constant characterizing electron donating or accepting push of substituents on the molecule. Then the reaction theory of Ingold about mesomeric and inductive effect, their influence on the reactivity of organic molecules. Hansch introduced the QSAR, and Hansch introduced also hydrophobicity substituent constants, or pi constant, characterizing hydrophobicity, or basically log P of substituents. Then ideas of Craig and Toplis, which form the basis of the early, but also the current drug design. I listed here some of these classical publications. Yeah, Craig, 1970, Toplis, 1972, Hansch, 1963. So these publications are more than 50 years old. I suppose none of you had a look or read these publications. But if you have time, I can really recommend you to revisit some of these really classical publications, because as I told you, these publications form the basis what we are doing now, what we are trying to now in the drug design and discovery. And finally, not so recent history, combinatorial chemistry, library design, and influence of building blocks or application of building blocks in chemical synthesis, because reactant used in chemical synthesis, this is nothing more, nothing less than just substituent connected at the end to some reactive group, reactive warhead. So here we have the, you know this already, here we have the most common substituents present in bioactive molecules, very common substituents, yeah, methyl, hydroxy, fluoro, methoxy, and so on and so forth. So what we need if we want to process substituents in computer? Of course, we need to characterize substituents by calculated properties, but we cannot use the characterization of substituents requires different properties than for the characterization of the whole molecules, because the main role of substituents is simply influence the properties of the whole molecules, not absolute properties by how they influence the rest of the molecules. Basically, these three, size, hydrophobicity, and electron donating accepting power, are the properties that are used most commonly to characterize organic substituents. Size, well, we can use volume of substituents. There are the sterimol, these are specific parameters characterizing substituents. I think there are 5 sterimol parameters characterizing different dimensions, width and height and other dimensions. But sometimes just the number of non-hydrogen atoms is sufficient to characterize the substituents. Hydrophobicity, also not so difficult to calculate. There are many fragment-based hydrophobicity contribution methods. Gaussian and Crippen is used most commonly, implemented in RD-Kit, for example. So one can easily calculate this hydrophobicity or pi constant for substituents. But electron donating accepting power, that's quite a challenge. One can find these Hammett sigma constants in tables. In largest tables, there are values maybe for 200, 300 substituents, these Hammett sigma constants. But it happened to me many, many times that when I was doing QSR analysis at Novartis, that just the value for the substituents, most active, most intrinsic, simply was not included in the table, was not available. So that led me to the development of the method to calculate properties compatible with Hammett sigma constants. The principle is very simple. We attach the probe, basically this is phenyl ring, to the substituents. For example, if we want to calculate Hammett sigma constants for pyridyl substituents, we will attack phenyl ring. And of course, the charges on this phenyl ring are affected by the substituents. It either pushes or pulls electrons from this probe, from these phenyl rings. And when we can do this for a set of substituents with well-known Hammett sigma constants, I used how many? 89 substituents. One can then correlate the charges with these Hammett sigma constants. Here we can see the statistical parameters. They are not so bad. Square of the correlation coefficient, cross-validated correlation coefficient 0.9188. It is even better shown on these two plots. There's the correlation between experimental and calculated properties. So here at the bottom, these are donors. At the top, these are strong acceptors. These strong acceptors, the situation is a little bit scattered. The points are a little bit scattered. But in this case, I would rather trust calculated value than experimental values, because it is well known that experimental measurement of strong acceptors is pretty difficult. And actually, everybody can calculate these Hammett sigma constants, again, on one of the tools on my website. This is the tool. So let's try to calculate Hammett sigma constant for some substituents. We need to mark connection of the substituents with the main molecule. So this is the substituents, and we will start the calculations. It was very fast. Here we can see the results. Sigma meta, sigma para, and additional as a bonus hunch pi hydrophobicity constants. So what has been done during the calculation? Smiles of the substituents has been sent to the server. On the server, instead of R group, the phenyl group has been added. 3D structures of this has been generated from the smiles. Then the molecules have been fully optimized using the XTB quantum chemical package. And then charges on this phenyl group has been got. And the sigma constants have been calculated using these equations I have shown you before. Let's try it for some other substituent. This will be strong donors, so Hammett sigma constants will be negative, yeah, -2, -4. Okay, now we have a method to calculate these Hammett sigma constants. Also, we can calculate also substituents hydrophobicity. So we can calculate these properties for a large set of substituents. In this case, we have the data for about 6,200 substituents. Well, let's do it interactive. So this x-axis, this is substituents hydrophobicity. On the right, hydrophobic substituents. on the left hydrophilic, and this epsilon axis, this is Hammett sigma constants. These blue substituents are color-coded based on the type of the root atom. In blue, we have donors. These are nitrogen substituents, and some oxygen, carbon substituents. Here we have carbonyl substituents, and at the top, we have the most strong known acceptors. And we can even we can simply browse through this substituent space, but we can also select subset of substituents. So let's say carbonyl substituents connected to the aromatic carbon. So this is the subset. These substituents in one cluster, one group, are similar, are bioisosteric substituents. Of course, the third dimension is missing. The size, we do not have size here because on the screen we have only 2 dimensions. So you have to consider size, well, in your head. Actually, when I was at Novartis, I developed something like this, a tool to automatically identify bioisosteric substituents. The procedure was simple: draw the substituents as we have seen before, and then make search in a database considering all of these 3 dimensions: size, hydrophobicity, and the Hammett sigma constants. And the chemists get list of similar bioisosteric substituents as shown here. Yeah? I do not have this yet on my website. I will maybe check it 1 month or 2 months, maybe it will be available. Chemists quite like these substituents because it helped them in their QSAR analysis. Okay, and with this, I came to the end of my presentation. I have here these QR graphs. On the left, this is link to the web tools. On the right, a list of my publications and some reprints to download. And I think we have quite some time. If somebody has questions, then I will be glad to try to answer this question.
No question.
So I have a remark maybe. So first of all, congratulations. I really love your work and your presentation here. I think that what you showed at the very beginning, the frequency of rings in bioactive molecules, right? This might change with time because some of the spiro rings that you showed before are now probably available in much more quantity that they were available before. So it might change then that they also get up in the more of the bioactive molecule space, right?
That is fully true. Yes, this is not written in stone. All of this is depending on the availability on the building blocks. Sometimes when a new drug will appear with some non-classical scaffolds, of course everybody tries to do the same scaffold or similarity scaffold, and then it really goes up. They zoom up. Yeah. Some time ago, with some Novartis colleagues, we analyzed how the popularity of rings develop in times. And one can see really very nice effects, dependence, on, as you said, yeah, on the availability of building blocks, some novel active molecules, or when some drugs is removed from the market. Yeah, so then the popularity goes down. This is very interesting, but also very, very complex process. Yeah. But as you said, this was just one spot in time. Yeah. So. Yeah, I would have a lot of publications on the any other questions. Yes, so.
I have a follow-up regarding these fragments. You show this color-coded by activity types, and did I correctly understand that the color means that this scaffold is exclusively for this kind of targets?
Yes. This means that all scaffolds shown are mixed, but if they have been color-coded, this is, I think I introduced more than 90% of molecules are, so this one can say exclusively on this active on the same type of the target. If active of many target, they have been gray. They have been
because I was surprised that some scaffolds which do not have even so many hydrogen bond donor acceptors, so they should be quite common across different types of targets, but they were mainly for GPCR, for example.
Or nuclear receptors, for example, they do not need too many hydrogen bonds. They just need some hydrophobic moiety going. This depends on the type of the target. Yeah.
Okay. Some other questions? Yeah. I saw a hand somewhere. No. Before he asked.
Okay. So Thierry,
Okay.
Thierry,
Thank you. Thank you once more.
This, okay. And this, it's already, it's running, okay. So good afternoon. I think I can start. Very happy to be here. My name is Thierry Langer. I'm a professor at the University of Vienna, head of the Department of Pharmaceutical Sciences, and it's my pleasure to be here and talk to you guys in this interesting meeting. And I will also afterwards give you a tutorial on ligand scout pharmacophore methods. So the topic of this speech today is I will try to explain to you our developments in next generation pharmacophore modeling and how we can apply this for molecular design. First of all, I want to thank a few people that are involved. So this is a snapshot of the people in my group currently at University of Vienna. But of course, all this work that I will be talking about is not only the result of these people, because I went to Vienna only 11 years ago. Before that, I was at University of Innsbruck and had a very nice group and a few people that I want to mention here that moved then also to the company that I created with other people together, Intelligent in Vienna, and still a lot of work is coming out of these groups. And I have also to acknowledge, of course, funding and collaborations with companies. Without them, we would not be able to do these developments. So, before I start, I just want to warn you, you can do a lot of calculations, but if you don't validate these calculations by experiments, they are more or less not so valuable, right? This is the so-called DMTA cycle. Sorry. Oh, I wanted to go back here. That is used in drug discovery, in rational drug discovery. You have an idea, you design something, you make something, you analyze, you test, and then you analyze the test result. And this is a cycle that goes and goes several times. And once you have optimized your properties of your molecule in all multi-dimensions that you need to have, then you can come up with a drug candidate and submit it to clinical development. And one of the crucial things at the beginning of such a program is to identify hits, to explore the hits, make leads out of them, and optimize the leads. And it's like finding a needle in a haystack, right? The drug candidate. And they will go back later to this image again. So important questions here that we are tackling with these methods are data analysis, which molecules to choose for biological assessment, how to modify molecules for optimizing the properties, how to build models for reliable medicinal chemistry decision support, how to design molecules that are void of toxicity risks. This is something that comes in later stages when you test in tox studies, and you might be thrown back again to the beginning when you find out that your molecules are toxic. And of course, it's not only data analysis, but it's also about data representation, how to guide the analysis of molecular modeling results, and how to make, for instance, use of MD trajectories for molecular design. And these are questions that we try to address with very specific tools that I will present to you now. A possible way to analyze interactions of molecules with each other, especially small molecules, but also bigger molecules with a target, with a protein, is to compare them in view of their preferences for specific molecular interactions. And you can do this, for instance, by annotate molecules with all interaction features that are theoretically possible. So we know exactly what molecules can do for interactions. H bonding, aromatic interactions, charge transfer interactions, hydrophobic interactions, coordination to metal ions, etc. And we don't know which ones are the important ones, but we can find out which one's the important one. For instance, if we have X-ray or cryo-EM or NMR structures of the ligands within the protein. In such a way, when we have this, we can then consider only those interactions that are really present in the complex. And out of the theoretical ones, there will remain only those who are really present. And these interactions here that I showed you are already encrypted in a so-called pharmacophore, right? So this is a pharmacophore that is derived now from an X-ray structure of a ligand in a protein. And we see that not all interactions that the ligand can theoretically do are really done in this complex. And the interesting thing is now how to find out how we can find this interaction. So just the pharmacophore concept is not something that is new. It has been out for a very, very long time. It dates back to Paul Ehrlich, basically, and people after him. And the definition of a pharmacophore is by Camille Vermouth, who was heading UPAC at that time and published in Pure Applied Chemistry in 1998. And it says, the pharmacophore is the ensemble of steric and electronic features that is necessary to ensure optimal supramolecular interactions of a ligand with a specific target and to trigger or to block a reaction, right? A biological response. This is very important. And when we look at these feature-based pharmacophores, we can say that these interactions can be encoded in features that encode electrostatic interactions, AIDS bonding, aromatic interactions, coordination to metal ions, halogen bonds, and so on. And we can derive them directly from an X-ray structure of a ligand with a protein. We can save them as three-dimensional objects that we can then use for virtual screening, for instance, and we can even project them on two dimensions to communicate with each other. Medicinal chemists, at least, medicinal chemists a long time ago did not like too much 3D. They liked to look at 2D molecules, and they discuss them always in the same way, and we help them with this representation. So they can say, okay, in the north of the thing here, I need a negative ionizable group that can accept some hydrogen bonds. On the south, I have a basic nitrogen that can be protonated, that will make also a charged interaction, and so on. So, in my eyes, the pharmacophore models is still probably the most suitable data representation method to guide medicinal chemists, because this is a language that unites medicinal chemists with modelers, right? And medicinal chemists already think in a way in a pharmacophore dimension. And when they talk about molecules, they can really explore this in this pharmacophore dimension. So it might sound a little bit old-fashioned, pharmacophore concepts, but it is not at all. You can push the technology of pharmacophores that is an old established technology to new borders, and this is just a snapshot here of the currently in development next generation of Ligand Scout. The software that I will show you afterwards, that shows here the X-ray structures of specific ligands in a kinase here, and you see with a click of a button you can generate the interaction pharmacophore models of these different ligands in the binding sites and use them later on for virtual screening, for comparison, for design. I talked about MD trajectories. In MD trajectories, there is a lot of information. It's not only the static thing, but also the dynamic of binding of a ligand to a protein. And we can transform these trajectories into pharmacophore space. And we can look at dynamic pharmacophores, how they change during time. For instance, how they are developed when a ligand moves into a binding site. Which is important, which are the features that are there all the time, which are the features that are not so stable. And with this information, we can derive a lot of additional knowledge for medicinal chemistry design. For instance, in this feature frequency time plot, you see here the pharmacophore features on top. Each little square here is a feature, so the two hydrophobics here, for instance. Green ones are the donors. And you can see in this plot here the frequency of the features. Some features are there all the time, some features come and go. And this helps us also to understand binding and to understand how to design better molecules. We can do this here, for instance, highlighting one specific feature and see in the trajectory where it comes and goes. The other one here is all the time there, for instance. This would be this hydrophobic here. This donor here is there very frequently. At a certain moment, it goes a little bit down. So this is information, and the other donor, this one, is a donor that is there, but not all the time, comes and goes, comes and goes. So this is something where we know then, this is a donor that is, for instance, not as important, and the other one, we should probably not replace it in order to keep the affinity. We also can look at the pharmacophores that are the principal pharmacophores that come over and over and over again. And this would be, for instance, a plot here where you see this pharmacophore with 6 features occurs like 37 times in these frames that we have generated. So this was a dynamics of a short MD simulation with 300 frames. This is probably more than 10%, this is 10%, and so on. And so you can see here the distribution of unique pharmacophores over the entire trajectory. And what you also can see, you can see which interaction is present in what % of the frames and which amino acids are contributing. So for instance, this hydrophobic interaction is 100% of the time with an alanine, 92% with another alanine, 100% with a leucine, and so on. So you can see the amino acids that are interacting with a specific interaction feature. So how to use this information now? And we thought about a way to help people with the analysis of the trajectories by creating pharmacophore feature vectors and compare them by building a hierarchical tree of these vectors. So each frame represents a vector in the space, and we can connect these vectors to each other. We can look which vector comes from a parent, a grandparent, and which kids are then coming from the vector. And this is a sort of analysis here where we can plot this in 2 dimensions. On the x-axis is the number of features. So this would be the feature that is common, the common ancestor in all pharmacophores, right? Then these are the one with 2 features, with 3 features, 4 features, 5 features, 6 features, and 7 features. And we can connect them and see how they are connected to each other. So when we click on a certain point here, we can track down where they are connected, which connections are occurring between these pharmacophores. And what we can also see by plotting the number of occurrences, making the circle bigger, we can see which pharmacophores are coming very frequently. This, for instance, and this one are the predominant ones. And here is also one that is predominant and a few in this area. So this was a work by Arthur Garot that was done in collaboration with Servier, and we analyzed together with Servier an application case that they were interested in a long time ago, was the analysis of binding modes of ligands in human glucokinase. And to see if there is a way to check if a molecule would be an activator of this kinase or an inhibitor of the kinase based on the behavior in the MD simulation. And by doing this, by comparing trajectories of an activator with a trajectory of a blocker of an inhibitor, we could see indeed that these dynamic pharmacophores have a difference. In the upper part, there's the one, the other one is in the lower part, and you can see here, the one is the activator one, this is the inactivator, oh sorry, the upper one is the inactivator, and the lower one is the activator, and you can see different pharmacophores in these plots. And when you plot them on top of each other, you can clearly see a difference here. The one activator and on the other side the inactivator will go in different directions. Now what you can do with these pharmacophores in these plots, you can go here for instance where there is a clear discrimination already between activators and inactivators. You can use such a model for screening and you will retrieve molecules that will inactivate the kinase. When you use these pharmacophores here for screening, and you can use it in parallel, you can screen with 20, 30, 40 pharmacophore models in parallel, you will find molecules that will activate the kinase because they have this binding mode pharmacophore for activation. Right? So this is a case study here that clearly shows that we can use this dynamic information for defining or finding molecules that will have 2 distinct different binding modes one activating, one inactivating a protein. Okay, so now let's go back one step. The software that is used for doing this is Ligand Scout. It is a fully integrated molecular design package nowadays. We have invested a lot in the graphical user interface. There is also a workflow integration in KNIME, in the KNIME workflow platform, and I would say it's accepted nowadays that Ligand Scout is the leading solution for Pharmacophore-based modeling, and we are currently developing the next generation capabilities, namely fast high-throughput screening on gigascale libraries, because, you know, as Peter showed before, the size of the libraries goes exponential, you know, enamine with the billions of compounds. So we also want to be able, with a very precise searching machine, to go into this scale, and also do some other things for ligand-based modeling, for instance, this interactive Pharmacophore-based clustering. It has been used in a worldwide user basis. More than 3,800 papers are published in different application cases here: structure-based modeling, ligand-based modeling, virtual screening for hit identification, fragment-based design, lead optimization, and so on. Protein-protein interactions, drug repurposing, also toxicity prediction. At the end, I will show you an example of toxicity prediction and profiling of side effects. Just one success story, a recent success story here that was communicated by Pfizer. They had an X-ray structure of a ligand that they were looking, or they wanted to find other ligands for this protein, and they built a structure-based model and screened their library with this structure-based model, and found a hit here in their library that was already pretty potent, 70 nanomolar. So for a hit, this is very, very good. And the people who did this study told us that they never had been any success with other virtual screening methods. Only the Pharmacophore was able to give them this hit here, and with one modification, adding a chlorine atom here, they got already a much better hit, and this was the lead compound actually, with a better selectivity towards other kinases that they didn't want to hit. This a further derivative of this molecule went then into clinics, and unfortunately it failed in phase 2. It didn't make it, but it passed through phase 1. So this molecule could still then be repurposed for other applications. Another recent success story, together with Edgar Jacobi from Johnson & Johnson, what we call Fragment Scout, is a workflow where you take X-ray structures of fragments that are co-crystallized, several on top of each other, and automatically generate a common pharmacophore of all the fragments that are found in these binding pockets. And then you run a virtual screen asking for a minimum number of features to fit and not all the features, so that you can then find in a binding pocket bigger molecules that would fit to these different fragment binding sites. And we experienced this on a protein of SARS-CoV-2, the NSP13 helicase. And this paper just came out a few months ago. So the starting point were X-ray structures of these fragments that were measured at the diamond light source and provided by X-Chem. For each of these structures, we created a Pharmacophore-based model, joined them automatically, did the virtual screening, and then we screened, or not we, but the guys at J&J screened their compound library for hits. And interestingly, this worked quite well. So we found, and these are just the binding sites here that were used. The nucleotide binding site was 13 structures were provided, 12 structures for this binding site here, and 3 structures for this binding site. I just show the example here from this with the tree structures here, just as the example. So you combine these models to something that is pretty complex, and then you ask the software to retrieve a minimum of a certain number of features. And then they went to a hit validation assay. So they had approximately 400 hits that were discovered from this 1 million compound library. And they were validated. 25% of them were active in one assay, 33% in the other assay, and 13 compounds were active in both assays with good selectivity indices. And these are now here shown in this table. And here you can see which binding site they were found. So most of them were in the stalk zinc binding site. One was found to be active in 2 binding sites, and the other ones in the RNA binding site, and only one in the nucleotide binding site. And interestingly, they had done the same experiment with the high throughput docking approach. So they docked the entire library using GLIDE, and they found 1 hit here with GLIDE. This was this J&J 1301 compound, whereas all the other hits came from the fragment scout approach in ligand scout. So this is a very nice validation story here. And you can do the same thing. You don't need X-ray analysis. You can do NMR structures. And this is a recent paper by Julien Ertl at University of Vienna, who did a similar experiment using NMR structures. So he resolves NMR complexes, transforms this into pharmacophores, does screening, and finds new hits. And this was in the KRAS protein. So you can also do something, generative design of molecules directly into a pharmacophore. This was done by collaborators in Japan here. They synthesized and tested compounds for the DDR1 protein. They wanted inhibitors here. And what they did is they trained a generative neural network to build compounds based on the learning of Campbell. So these are compounds that are bioactive, that have been synthesized, have been tested, so somehow privileged structures. So this agent knows how bioactive molecules look. And then in the next phase, you give a pharmacophore to the generator and ask to score compounds that fit well to the pharmacophore high and compounds that don't fit to the pharmacophore low. Then the system learns, and after a while, the system generates molecules that fit very well the pharmacophore. So they ran this, and at the end, they also made a binding affinity score, and they synthesized the top ranking solutions here and came up, the best compound had a 2-digit nanomolar range in IC50. This is this compound here from scratch, just starting with a generative AI model that is instructed with a pharmacophore fit score. Let's switch gears now. We also do open source tool kits here, and the one that we are using at the university is our CDP kit. So this is something in an analogy to RD kit with the advantage that it has the full 3D and pharmacophore kind of interpretation, virtual screening capabilities in the toolkit. So you can use this for free. There is a website here, the cdpkit.org website, where there is the documentation, where there is a cookbook, where you can look at the libraries, download them, etc. So it's implemented in C++. This is basically the work of Thomas Seidel and Oliver Wieder. It contains all basic functionality for cheminformatics workflows, substructure search, pharmacophore search, etc. Comprises the chemical data processing library, which we call CDPL, together with command line software tools. So there are binaries also there that you can directly use in scripts But you can also, of course, use this in a C++ API or in a Python interface to write your own piece of software using these libraries. So we have integrated also major machine learning libraries such as scikit, PyTorch, and TensorFlow. And this represents at the moment our standard environment for prototyping new algorithms, and the follow-up implementation then goes into LiganScout and 9 platforms. But again, the thing here is open source, so you can use it yourself and continue develop also, and there is a GitHub webpage for this. Now let's go back here to this question. The big haystacks, right? So people claim now that you get better hits if you get a bigger haystack. There are some papers who claim this. So for this, of course, we need, if we want to have high precision Pharmacophore search, we need to enumerate the compounds. And Peter said already in his lecture, this is a problem with the number. So because these libraries, when you have 3D conformations, they can become pretty big. And of course, you need also something, a conformer generator that is really fast if you want to enumerate billions of compounds. And you also need faster alignment procedure, of course. And so we have been working on that. And one thing here, the question is, of course, how big these libraries should be. And when we look at a few numbers here, the chemical space could be 10 to the 90. Some people say up to 10 to the 120. Typical hit rate that you get in screening is around 0.1%, so 1 hit out of 1,000 compounds, typically, maybe a little bit lower in virtual libraries, but still, even if we go to the 0.1%, you would get 10 hits out of 10,000, 100 hits out of 100,000, 1,000 hits out of 1 million, and continue to think. As if you screen 1 billion, how many hits you will get. So this becomes unmanageable. So you need something. The question is really, is this smart to go so big, or is it not better to restrict yourself to compounds that are like privileged in a way, and that you can synthesize and modify in an easy way? So I believe that the numbers game is not really the game to play. I really believe that maybe a few 100 million compounds might be enough. You don't probably not even go to the billion if they are well selected. Still, if you want to enumerate a few 100 millions, you need a very fast conformer generator, and this is also a contribution of Thomas Seidel here and a validation that we did together with some other people here in our group. It's Conforge. This conformer generator has been shown to be probably the best performing, best high quality open source conformer generator by now. It has shown that it's especially valuable for complex structures such as macrocycles. And we are, of course, integrating this also in the next generation of LiganScout. And together with the new alignment that we have built in, this is the basis for Pharmacophore-based gigascale virtual screening. This is the algorithm that we have published for the alignment, for the Pharmacophore alignment, with a big difference to the previous method. It's based on iterative improvement of heuristic guesses. So it tries to match the maximum of the features. It's much faster than the previous methods, and you can tweak it for speed or for quality. So if you have a small data set and you want to go for the highest alignment quality, you will set a higher number of heuristic guesses. If you just want to go for speed and you don't care if you miss a few compounds, but you want to be very fast, you can go low. And the great thing is you can ask for a number of features that are optional without telling which feature you need to be optional. So you can have a model that has 30 features or 40 features and ask for a minimum of 6 or 7 or 8 features, and the speed will be not slower than, or not much slower than if you would ask for a total match. Right? And this is the big advantage of this algorithm, which makes it an alternative to molecular docking now. Much faster, but the same quality actually. When you generate pharmacophore models in empty binding pockets. Not only the pharmacophore that is there from a ligand with your protein, but also all other features that could be there because amino acids are on the surface of this protein's binding site and are waiting for some interactions. With generating an apposite pharmacophore model, you can generate these interactions and then you can screen with this new algorithm. And it's like probably 100 times faster than the fastest docking engine. So where do these apopharmacophores come from? This apopharmacophore method comes from what we called the GRAIL descriptors. GRAIL is the abbreviation for grids of pharmacophore interaction fields. And this is also published, a paper that is out there since a few years and that we now use to derive pharmacophore models in empty binding pockets. From X-ray structures, but also from MD simulations. We can run this on MD simulations. And what is done here, you define a grid in your binding pocket, and at each grid point, you calculate not an energy, but a probability for a specific pharmacophore feature. And we have a dozen of pharmacophore features, you know, H-bond acceptor, donor, hydrophobic, positive ionizable, negative ionizable, halogen bond, etc. And for all of these features, at each point, there is a certain probability for a pharmacophore interaction. If there is a partner, a binding partner, in the right distance, in the right angle, for instance, right? And so each point gets a score, and then we can go from the highest score to a certain threshold, and we can ask, let's put the features on these points that have the score in a certain threshold. And in this way, we can generate pharmacophore models that are independent of a ligand, that only represent the wishes of amino acids in a binding pocket to interact. And this is here an example where we can contour these plots here, and there is, for instance, for this phenylalanine 130A in the HSP90 case study, you can see here the areas where there would be aromatic interactions, areas for hydrophobic interactions, areas for positive charge, pi cation interactions. Here, this is a negative charge interaction with a positively charged leucine residue, etc., etc. So you can get this, and at the points with the highest score, you can put the features. So how can you use this? You can use this to, again, get medicinal chemistry design guidance. You can focus on specific regions. For instance, you can look at water molecules in a trajectory and find out, is this a water molecule that is happy because it really makes good interactions and wants to stay there and stays there in the trajectory? Or is this a water molecule that always gets exchanged by another one because it's entropically disfavored, right? And so we know exactly, for instance, which water molecules we want to replace. And this is this typical magic methyl concept in medicinal chemistry. You know, sometimes at the end, when you have already optimized your lead compound, you will go and place methyl groups here and there and check if you could probably enhance the affinity. And this is the so-called magic methyl, and it's based on entropic contribution because just a little pocket with one or two water molecules gets filled by a methyl group, and then the affinity gets higher. So this, again, is the basis also of the development of additional descriptors that I show you now in a minute. This is the work of Christian Fellinger here, that is also just published in JK-Min's model. He used these GRAIL descriptors together with scalar descriptors to train a machine learning network to predict binding affinities. So he used the GRAILs of the PDB bind, so he calculated all GRAILs for all ligands in the PDB bind, and developed a model where you can now then put a new ligand in this protein, and with these gray descriptors have an affinity prediction of this new ligand. You can use this for affinity prediction, you can use this for post-prediction, for docking, right? Because the most favorable energy, of course, will be there. You can use this also for QSAR, etc. So check this out if you want. This has been validated in several studies so far, and is an extension, in my eyes, of 3D QSAR methods. What you also can do is you can create a hybrid framework where you combine these voxel-based pharmacophore maps of the protein binding site with a graph-based representation of the model. And then process these grails with a 3D generative convolution network. And this then can serve as a context vector for GNN interpretation of the ligand. And what we found out is that the performance is comparable to state-of-the-art methods, but the interpretability is much, much better because we can really see which parts of the molecule contribute to affinity by just looking at the gradient distribution. So we can find out by this method which part of the ligand is highly responsible for binding affinity. You can also do this with DFT methods, quantum chemical methods, but the calculation is way, way, way more complex and longer. And there is a preprint just put out here on ChemRIF, ChemRxiv, this ChemArchive. Daniel Rose and Thomas Seidel did this. You can download the paper here. It's in review at the moment. And you can download the code and run it yourself. A pre-trained model. And what you see here, what is done, you have this protein ligand complex. You have the Grail map. And the ligand is encoded with this GNN encoder with a multilayer perceptron. Here the Grail map is also encoded in a CNN and makes the pocket representation. And then we can calculate this model attribution with integrated gradients and predict PK, and also see which region here is important for the affinity. This is how it works. It has been calculated on PDB-Bind on the version 2020 with 19,443 annotated protein ligand complexes. We did a typical machine learning sync core split, 290 test samples, and we did an LP PDB-Bind split with 4,860 test samples. And I don't want to go into the details here, but what you can see is clearly different channels here for aromatic interactions, H1 donors acceptor interactions, positive negative ionizable interactions, and you see here these are the channels and these are the model attributions that will then tell you which part of the ligand is important for which kind of these interactions. And yeah, this is detail here. Here is the evaluation, performance evaluation on this CAS 2016 core set. And the model here, Griffin, performs on par, a little bit worse than the best one, but on par of or better than a lot of other models. And it's extremely fast. And you can also give it a try. So benchmark data is available for testing. You can try it with your own actives and inactives, and just let us know if you have specific ideas for workflows. We are happy to collaborate on this. So back to the haystack again here. Is there a way to do high quality 3D virtual screening without alignment? So, you know, there's still the process to align each conformation of your ligand to the Pharmacophore model. Is there a way to avoid this? And also, we have presented here a way where we can do this. Daniel, also in his PhD here, So when you do a classical way, you have the ligand database, conformers, each conformer is annotated with a pharmacophore model. You have the pharmacophore that is either derived from an X-ray structure or a ligand-based model. In this case, it's a structure-based model, and then you align and you get your hit list. Question is now, can we do this faster without the alignment? Yes. So what we do is we have the ligand database, we enumerate them, we can generate the conformations, we can generate the pharmacophores, and then we encode them into vector representations. So we get a pharmacophore vector database, and then we use the same technique here for the one vector that is represented by the structure-based pharmacophore query. And then using embedding space property, we can generate a hit list of the pharmacophore vectors that are most similar to the query vector. And we don't need to do any alignment. So there's one step in the preparation that takes longer because we have to calculate the vectors. But then, when we have all the vectors, the screening is much, much, much faster. And this vector embedding space concept basically is the concept that vectors pointing in the same directions have the same, encode the same properties. For instance, when we think about data science and computer science, these vectors point in the same directions. When we talk about a molecule and caffeine, they point into the same directions. And when we talk about cheminformatics, this should be pointing in between because this is the same data science, computer science that is about molecules. So this is this vector embedding thing, and it's often used in this REX concept, which is the retrieval of concept in database. It can be used in search engines, in recommender systems, also in chatbots. And we use this vector embedding space approach for our pharmacophore thing. So what shall we compare to train a pharmacophore embedding model? So what we do is we have a query pharmacophore and we have a target pharmacophore. So this would be the query pharmacophore. This is the target pharmacophore. And then we know there is a supergraph, a subgraph. The query pharmacophore is a subgraph of the target pharmacophore. And in this vector embedding, in this target embedding region, we see here, this would be the target region. And we just need to look for all vectors that point into this region. Ones that are off would not be considered. The query embedding is here, and the dimension that is opening up the space from this query embedding is the target region. And all the pharmacophore vectors that we find here should be interesting ones, right? So what we do for this learning, we used a contrastive loss model that use shared weights between the encoders here for these vector embeddings. This is just a bit of detail here for this technique here. And again, here is the representation, the subgraph that we have here as a query, one supergraph that fits perfectly in the target region, and another graph that would not fit and would be in this dimension, would be somewhere else, right? And therefore not in the target region. It works reasonably fast. So we have done this on a data set of Didier Rognan and colleagues with one query, 3,920 ligands that had all up to 25 conformations. On the disk, this database took 50 megabytes. With the pharmacophore vectors, it takes 70 megabytes. The screening time with CDP kit is 2 seconds, and with the pharmacophore match is 0.02 seconds. It's a factor of 100 faster because you don't need to align, right? So now this becomes really also amenable for ultra-large screen ultra-large library virtual screening. And what we also can do is we can still have potential for even faster database search if we use vector database formats instead of relational database. So this was I showed before was a relational database, but we are working on applying this concept using a vector database for even faster retrieval, because then we just go entry point in the vector and can be very fast in this vector database. Last thing that I want to stress today is the application of pharmacophore models in late stage, I would say before clinics, but even when we have the models in the design stage. And this is about de-risking neurotoxicity. So these are the results of a European community project, IMI2 initiative, that I was leading at University of Vienna and this consortium. And we had submitted a proposal that was then considered and funded to address 3 major problems in neurotoxicity, namely seizures, psychological and psychiatric outcomes, for instance, suicidality, and the third one was peripheral neuropathies. These are all problems that are poorly addressed with animal models. So predictive animal models are not really good in this. They become better, but of course also in the aim of replacing, reducing animal experiments, having these in silico models will help. And so in such an approach where you have toxicities, there are 2 main possibilities to go for. The first one would be the target-based approach, when you know exactly what molecular initiating event is responsible for a specific toxic outcome, right? For instance, seizures, we know if we block the GABA chloride channel, we will have a seizure, basically. And therefore, it's quite easy, and there are X-ray structures, cryo-EM structures. So for this, for instance, here, we can do structure-based modeling, we can generate pharmacophore models that will predict if a ligand will bind and block this channel or modify the conformation of the channel in a way to have a negative allosteric modulator, for instance. For other things like suicidal ideation, this is much more difficult. There is no real way to know what the molecular initiating event is that some people kill themselves when they are on certain drugs. And this is unfortunately a reality. And pharma companies are very much worried about this, of course. If you have a drug on the market and it turns out that people kill themselves when they are on this drug, this is a disaster. And one of the compounds that was the basis for this program was rimonabant. This was a drug that was brought to the market by Sanofi. In Europe, in the US, it was never approved. Was a drug that should help to lose weight, but instead of losing weight, the people would just throw themselves out of the window and kill themselves. So this is a big problem. And the third thing is, of course, the neuropathies, peripheral neuropathies. Most of the cancer drugs will cause pain in the periphery. And this is also something that we need to know why. So again, here for the seizures, it was quite simple, I would say. We can generate pharmacophore models that predict if a ligand will bind to this channel and block it. For neurotoxicity addressed with suicidal ideation, what we did is we looked into adverse outcome databases. So which drugs have been reported with suicidal ideation or with committed suicides. And there is a huge list of drugs that are annotated. So you can look at these drugs, you can try to find out what is similar in the structural motifs that they have to elicit suicidal behavior. But of course, the problem is the negatives. You don't know the true negatives, right? Which ones, how can you discriminate? And luckily in this consortium, there was a company that had developed a biomarker for one specific mechanism of suicidal ideation, namely the modification, post-translational modification of RNA that encodes for a serotonin receptor. So we know that certain drugs will enhance the post-translational modification of RNA that encodes for 5-HT2C. Once this happens, we know that serotonin all of a sudden doesn't have the affinity anymore to the channel, and people get more depressed and kill themselves. And we have clear signatures for this attributed with drugs. And this company had tested 1,800 drugs that are on the market and had a very clear profile for all these molecules. And we could use this data to train our models. And we then finally ended up with 7 models that clearly predict suicidal risk of compounds. We have 23 models for the GABA receptor, which indicates blockage of this channel. AMPA is there, NMDA, glycine receptor. 15 models for peripheral neuropathies, and we also have 3 models that will predict if a compound can go through the blood-brain barrier or not, because of course it's important. Compound needs to be in the brain. If you have a compound that can elicit suicidal behavior, but you put it on your skin and it doesn't penetrate to the brain, there's no risk, right? So you need also to know this. And so this whole thing was implemented in a NIME tool, actually NIME. There is this intelligent neuro de-risk profiler that contains all the models that are in this consortium, plus the conformation generator of Ligand Scout, plus the alignment of Ligand Scout. So you just have to plug in a list of smiles or an Excel table with your molecules that you want to analyze, and you will get a heat map that will show you which compound has a risk of a certain neurotoxic outcome. So with this, I'm almost at the end. I believe that pharmacophores can still be a source of inspiration, can still be something you do research on to develop new technologies, better, faster technologies. It is highly useful for hit identification and prioritization. It is also amenable for high precision virtual screening in exascale libraries. So we constantly now screen in the 3-digit million screen space. It's very comprehensive for medicinal chemistry guidance, and you can take into account dynamic effects by using these MD simulations and translate them directly into design. And finally, with being able to predict toxic outcomes, we get better lead structures void of toxicity risks. So there is still a lot of potential for this. So again, the standard pharmacophores are highly useful for hit identification, but when you go to a more detailed thing and you take into consideration dynamics, you have a highly useful tool for lead structure optimization and de-risking at the end. And with this, I want to thank you for your attention and just mention here the websites of the group in Vienna, the website of the toolkit, and the website of Intelligent that makes available Ligand Scout and continues to develop Ligand Scout. And I want to thank you for your attention and ready to ask your questions.
Thank you very much for the enjoyable talk. I just have a question concerning the encoding of the pharmacophores for the vector-based approach you have. How do you handle the Cartesian coordinate problem? So when encoding the pharmacophore, it's somewhere in space, and that can be in different points in space. How do you handle that?
Actually, this is pretty easy because we don't, we take a radial distribution function, actually. So it's coordination invariant. But very good question. Yes, Peter, you had a question? No.
Some other question? More questions will be during the tutorial.
Yes, I guess so. So I have a question. in this challenge, are there X-ray structures of ligand protein complexes?
Yes.
Also, okay.
So yeah, during the challenge you will be able to apply all these tools which you will learn during the tutorials, talking, USR modeling, pharmacophore modeling, and in previous challenges, in some cases, pharmacophore models get on top. So Who knows?
I'm curious this year how it will be.
Quite random and difficult to predict in advance.
Great.
So no questions. Okay, so thank you once more.
Thank you.
And we have a coffee break and we have half an hour before the tutorial. Please make a good right now. Yeah. Disable. Yesterday it was 250. Today I didn't check, so I don't know. Wow. I don't know. Wow.
And have they, do they know already what the challenge is?
No.
No, okay.
The target is still not published and the data is also published. So I will make them publicly available in just 15 minutes.